Good morning, everyone. I think I'm audible to all of you. So let us start. Topic of discussion is the biotechnology, and some of the portion we have finished last day. Audible, good. And discussion was the basics of biotechnology. Okay, discussion was basics of biotechnology. Okay, basics of biotechnology. And last discussion I was doing about the nucleus and chromosomes. So I told you last day, guys, nucleus have a chromosomes. What you have seen in the previous lecture, nucleus have what? Chromosomes. So within the nucleus have chromosomes. Okay. So within the nucleus have chromosomes. Then chromosomes, I told you there are 23 pairs in the human being. So what about number of chromosomes in human being? 23 pairs. So this one you will know very well. 23 pair chromosomes. And these chromosomes are different types. Even the one father and second is mother. Both have a 23 pair of chromosomes. So one side is the father and second side is the mother. Both have a 23 pair of chromosomes. Okay, 23 pair of chromosomes. 22 pair I told you last day. It's a XX autosomes. And one pair is the X with Y. But chromosomes always in the pair. So 22 pair in the father, X and X pairing, and one pair is the X with Y pairing. It is the condition in the case of father. And what about mother? Mother have each and every chromosome X type. 23 pair X. This is what? 23 pair X chromosome. Okay, it's about 23 pair X chromosome. Okay, X chromosome. Later on, I discuss with you guys about cell division. About cell division. In the case of cell division, I told you through two different phases cell division happen. The one is the interphase. Interphase. And second is the M phase. Interphase, you people have seen it's a pre-division phase, a regulatory phase, and M phase is what? This is a division phase. So here the cell divide accordingly. Interphase is more important. Last day I told you because it's a pre-division or regulatory phase. So every regulation and control of division is depend on interphase. And even the cancer type of problem I told you, it is abnormality in the interphase, because M phase is just a response phase. Here cell divide and redivide. Here cell divide and redivide. And here M phase, you will get it. It's a kind of division phase. And then told you there are two types of division. The one is called mitosis division and second is called as a meiosis division. So there are two types of division. The one is mitosis division and second is meiosis division. And two more discussion, even information I told you. Mitosis division, this is in somatic cell. Meiosis division perform in germ cell, generative cell, or reproductive cell. So germ cell, generative cell, or reproductive cell. So mitosis in somatic cell and meiosis are in germ cell, generative cell, reproductive cell. And then last discussion I told you, if a parent cell is 2n type, after division it forms 2n and 2n. Same number of chromosome, mitosis. If parent cell is 2n type and after division it forms half and half chromosome number n and n. So it will be the part of meiosis. Now you come. Last day we stopped here. Good morning, everyone. Last day we stopped here. Actually, what are the reproductive cell? I hope you know very well. And somatic cell, the location I'll tell you. Reproductive cell, everyone know. Inside the father side, testes. Inside the mother side, ovary. So what about the reproductive cell, germ cell, or generative cell? So it's very common and everybody know. Germ cell, generative cell or reproductive cell are what? Father side testes and mother side ovary. It's a reproductive cell. So father side testes and mother side ovary, it is what? It is a reproductive cell. Okay, everyone. What about the somatic cell? Listen, it's a very common sense. A part of reproductive cell, rest of all body cell is the somatic cell. Common sense. So no need to mug up it, guys. If reproductive cell, you know, yeah, definitely everybody know reproductive cell. Father side testes and mother side ovary, it's a reproductive cell. And a part of these reproductive cell, rest of all body cell is the somatic cell. Rest of all body cell is what? This is the somatic cell. Okay, everyone? Fine. Rest of all body cell is called somatic cell. Now come. Okay, now come another one. Rest of all body cell is called somatic cell. Example of somatic cell. So a part of testes, yeah, ovary, any body part you take, hair, nail, blood, bone, muscle, skin, everything. A part of testes and ovary, it's a somatic cell. So I think somatic cell, no need to write any examples. A part of reproductive cell, all the body parts are what? Somatic cell. You people can understand. You people can understand easily, guys. Okay, you people can understand. Now, what happened in the case of mitosis division? Achha, last day I did not tell you. N represent as a haploid in biological science. Haploid means chromosomes are not in pair. 2n is equal to diploid. Diploid means chromosomes are in pair. So we human being is what? 2n nature. Our chromosomes are 2n nature. Means our chromosomes inside our body is a diploid. Means in pairing. Just like we have a 23 pair chromosome. Just like we have a 23 pair chromosome. This is the concept. Okay, just like we have a 23 pair chromosome. So we have a 23 pair chromosome. 2n. We are 2n nature. Diploid. Okay, 23 pair chromosome. Now come. If a parent cell is the 23 pair of chromosome number, it divides from 2 cell, 4 cell, 8 cell. But the number of chromosomes same as a 23 pair and 23 pair. So it is what? This type of division is mitosis division. And this type of division where perform? Somatic cell. What happens if parent cell is a 23 pair of chromosome? After division it become 23 and 23. Number of chromosome become half. It's a meiosis division. And inside a reproductive cell, okay, whenever the sperm and egg formation happen, in this period of time, testes or ovary involved in meiosis division for sperm and egg formation. That is the reason when the sperm formation happen, sperm have hardly half number of chromosome, 23. And similarly, egg have only half number of chromosome, 23. So those sperm and egg have a 23 and 23 chromosome, half number of chromosome. It is because of these sperms and egg produced from the testes and ovary. So this is the reason meiosis perform here. So this is the reason meiosis perform here. Okay, meiosis perform here. Now you just see about the importance of these divisions. Suppose we are here, guys. We all are here. It's a father. Father means 2n. It's a father. Father means 2n. It's a mother. Mother means 2n. It's a father. It's a mother. So father means 2n and mother means 2n. Okay, everyone. See, 2n means 23 pair. 2n means 23 pair. And this half, I can tell you, this is the n. This half is the n. This half is the another n. Similarly here, this half is the n. This half is the another n. So n and n, another n. So this half and this half is what? This is a haploid. This half and this half is what? This is a haploid. Okay, haploid. Now listen what happened, guys. When the parents involved in reproduction, see here. When the parent involved in reproductions, so which body parts involved into the reproduction? Definitely testes and ovary. Testes and ovary. Okay, testes and ovary. What happened in this case? Very simple, guys. This testes and ovary involved in reproductions. So reproduction means it will become half and half chromosome. And what happened from the testes size? Sperm formation happened. And from testes to sperm formation, it's a reproductive cell. The reproductive cell divide. Which type? Meiosis division. And that is the reason half number of chromosome produced in the sperm, either this N chromosome or this N chromosome. And similarly from the female side, either this N chromosome or this N chromosome produced in the egg. Now it is depend on the combinations. Now it is depend on combinations. If father side, this N is coming in the sperm and mother side, this N is coming in the egg, and now have a fuse and it's a 2N. 2N means it's a zygote. Again become 23 pair. And 2N zygote is what? All set are X and X. So definitely it will be a daughter. Definitely it will be a daughter. If father side this sperm will fuse with this combination of egg. Again 2N. Again a whole set is X, whole set is X. So again it's a daughter. And what about the other possibility? If father side this XY wala set come, okay, and mother side this X set come, So it's a 2n again, but here Y is here, so of course it's a son. If father side this n combination and mother side this n combination, so it's again son. So ultimately what happened during the reproduction, guys? In this combination we all boys and girls form. In this combination we all boys and girls form. You see the results. You see result, guys. In this combination we all boys and girls form. Okay, in this way, guys, we all become again 23 pair. We all again become here 23 pairs. Now you just come, guys. Those guys, you people are listening me. Agar you are son, so you are this combinations. If you are a daughter, female, so you are the this combination. So ultimately what you got? Maleness and femaleness, it is totally depend on father. Female side have not responsibility for the baby will be a son and daughter. Reason, it's entirely depend on father. If father side this set of chromosomes are coming, this set of chromosomes are coming, it's a daughter. If father side this set of chromosomes are coming, okay, it's a son. Because female side have only S and X. So if this type of question anywhere by chance ask you as a information, I'm telling you, it will be a male or it will be female, only determined by father. Mother is not responsible for the determination of sex as a male or female. So only father is responsible. This XX set will come, it's a daughter. This XY set will come, it's a son. Simple. Now what is first day? We all people were the zygote. We all people were the zygote. Suppose this is a son, and this son is going to develop. Okay, this son is going to develop. How the son will develop? My question is, guys, this is zygote. This is zygote. My first question, ready to give me answer, guys. Tell me, zygote is a somatic cell or reproductive cell? Tell me, zygote is a somatic cell or reproductive cell? If somatic cell, so this type of division property happen because the zygote will develop into the mother body, and in 9 months and 6-7 days we become as a newborn baby of 3 kilogram. So you tell me first, this zygote is a somatic cell or reproductive cell? If you will identify zygote is a somatic or reproductive cell, so definitely you people know very well somatic cell have this division, the reproductive have a this division. So finally you will understood how zygote will develop inside the mother body through mitosis or meiosis. Diksha, twin baby wala concept, I'll come there. Before going to start the cloning na, I'll discuss with you about the twins. Wait bacha, wait. और अनिल ये क्वेश्चन एनडीए में आया है यार, इससे बाहर कुछ नहीं आता, ट्रस्ट में, क्योंकि वो सारे भी पेपर्स यूपीएससी ही बनाते हैं. Great. Everybody given me satisfactory answer. Okay, what about the satisfactory answer? You all people respond me, it's a somatic cell. So nice. And you people 100% correct. You people are 100% correct. Okay. So you can connect it. It means zygote is a somatic cell. So definitely people started to give me answer. It involved in mitosis division. It involved in mitosis division. So now you come here, guys. Now come here. Now come here. This is what? This is what? This is zygote. This is zygote. Okay, and now zygote will develop through mitosis. Okay, during gestation period. During gestation period, when people are developing into the body, during gestation period. So which division through gestation period? It's a mitosis. Okay, through mitosis we are going to develop. It's a gestation period, and finally we become as a 3 kilogram of baby. And finally here we become as a what? As a 3 kilogram of baby, a newborn. Suppose newborn baby born. So newborn, okay, newborn baby born. So newborn means new baby born. Okay, and now one thing more, guys. This is the reason when a 3 kilogram of baby born, each and every cell have a same number of chromosomes. Reason, it divides through mitosis division. And now this 3 kilogram baby is in front of me, and they become as an adult. Like me, you, we all people are the adult. Inside the adult, when you people were doing development, so inside your every cell have a mitosis division. Okay, and now finally you become 50, 60, 70, 80 kilogram. Okay, it's what? It's because of mitosis division. Okay, it's because of mitosis division. So because of mitosis division, you become adult now. Okay, adult now. So inside your body, entire body have a which type of division? Which type of division? Mitosis division. Now I have a question, guys. In this 3 kilogram of baby, in this 80 kilogram of baby, 80 kilogram of adult, 80 kilogram of adult. Okay, each and every cell have same chromosomes. And you know which chromosome? This chromosome. Those chromosomes first day you got from your papa and mama, 23 and 23. In this adult, each and every cell have a same chromosome. 100% same chromosomes. Actually, inside this chromosome have a presence of DNA. In this adult, 80 kilogram, okay, each and every cell have a same chromosome, and within the chromosome have DNA. This will be the next discussion, because now I am discussing about chromosome. After it, I will discuss what are inside the chromosomes. If chromosomes are same, so definitely DNAs are same. And this is the DNA, those similarity we are going to check. In the case of DNA fingerprinting. Suppose I did some crime, okay, and my hair drop here, my saliva drop here, my sweat part drop here, anybody's semen drop here, anyone. Okay, now from this semen, from this saliva, from this hair, from this blood, okay, what are there? Inside it have their own cells. Within the cells, what are there? Nucleus. Within the nucleus, what are there? Chromosomes. And what are inside the chromosome? DNA. Okay, if there here is a DNA, okay, here you fall, here fall here, so definitely DNAs are there. And you match with the suspect, guys. Those body DNA will match with it. Definitely that person was there. Even in the absence of eyewitness, through the help of DNA fingerprinting, you people can identify in the forensic science. So same, each and every body cell have a same chromosome, so each and every body cell have same DNA. This is logic, and I hope you people remember it. So why DNA fingerprinting become possible? Here you should thanks to mitosis. Each and every cell have a same chromosome. So same chromosome, same DNA. And this is the reason through DNA fingerprinting, if we people will take any of the cells, your each and every cell have same chromosomes, and definitely you will identify. And inside your each and every cell have what? Those chromosomes, your DNA you got, 50% you got from your papa and 50% you got from your mama. And that you people know very well. Okay, this is the reason if you want to match your chromosomes DNA, within the chromosome those DNAs are there, so 50% papa and 50% mama will match. And now it will be proved that you are the son and daughter of your parents. So through the help of DNA fingerprinting, we can do a lot of analysis. But this time I'm not discussing DNA fingerprinting. But I'm very sure you people understood why each and every cell have a same DNA. Reason, each and every cell have a same chromosome. Reason, mitosis. I want everyone answer, yes sir, I understood. Why DNA fingerprinting? Okay, possible. Okay, and the reason I also know very well why each and every cell have a same DNA sequence. Okay, each and every cell have same DNA sequence. Okay, each and every cell have a same DNA sequence. Okay, each and every cell have same DNA sequence. This is important. This is important. Great. One more concept I want to discuss with you. Because DNA fingerprinting, cloning, entire discussion I'll do in the biotechnology. But this is the basic, guys. And this type of question is a UPSC demand. UPSC never has the technical part, only basic parts. Only very, very basic part that UPSC has. So everybody clear. Now one more concept I want to give you. One more concepts. And the more concept is the cloning. Okay, cloning. Cloning I'm not describing you. The basics of cloning I'm discussing. DNA fingerprinting I could not describe. Just basics of DNA fingerprinting. So at least you understand why in each and every cells have a same DNA sequences. My statement is about cloning. So my statement is here about the cloning. Okay, my statement is here about cloning. What is cloning? Same multiple replica. You know guys, my simple question is, if I want to develop a clone, if I want to develop a clone of you people guys, so tell me, you people will give me your somatic cell or reproductive cell? If I want to develop your clone, you guys will give me your reproductive cell or somatic cell? Tell me. If I want to develop your clone, so you guys will give me somatic cell or reproductive cell? Try to understand. There is a lot of logic here. If I want to develop a clone, so you people will give me your somatic cell or reproductive cell? Fast. So you guys will give me somatic cell or reproductive cell? Somatic. People are telling me somatic. And here I will tell you guys, you people are 100% right. Because somatic cell divide through mitosis division. If reproductive cell you will take, so ultimately your number of chromosome become half. And you people develop because of 23 pair chromosome, not half number of chromosome. Okay, so definitely you people right. If you want to develop a clone, then everybody give me somatic cell. Okay, reproductive cell, if you will give me, the reproductive cell divides to half. It will become half number of chromosomes. And further, this half number of chromosome 23, how it can divide? It's odd number. And you people know very well, here in the case of zygote, when the zygote develop a baby, it was a what? It was a somatic cell. And somatic cell divide through mitosis and then the entire body develop. So if I want to develop you people guys, but not inside the natural condition, some artificial condition I adopt, I want to produce you. So ultimately as a zygote is a somatic cell, so any of your body part somatic cell you will give me. And some time of some chemical may I will adopt this type of procedure and finally you people will develop. How it happen, it will discuss under the cloning chapter. How it will happen, this discussion I will do in the cloning chapter, not here. So I think somebodies are confused, but they understood, yes sir, actually in the case of cloning we will take a somatic cell. Neither somatic cell, if we will take a reproductive cell, the reproductive cell cannot go in the mitosis and not develop entire body. This is the problem. Okay, this is the problem here in this case. Okay, this is the problem here in this case. Now come. I'll give you some examples in the proof of cloning. You know the first cloning was of frog cloning. The first animal cloning was of frog cloning. The first animal cloning was frog cloning. But first mammal cloning, I hope you know very well, it was a sheep. It was a sheep. Okay, it was a sheep. It was a sheep. And this was a male or female. I hope you know very well, it was a female sheep. Okay, it was a female sheep, that clone was developed. So the first clone was a female sheep. And name you people know very well, its name was Dolly. My question is only here guys. Maybe somebody will give me the correct answer. Which body part of this sheep even used? Which body part was developed especially for the formation of clones? The somatic cell, though you people know very well, definitely somatic cell will be used for the formation of clone. My question is, which body part somatic cell was used? Anyone know? In the case of cloning, I will discuss guys, but I am giving you lot of basics. I am giving you lot of basics. And this is very, very important here in this time. Okay, this is very, very important here in this time. एक कैसे लोगे? एक ऑलरेडी वो क्या है, एक हैव अ हाफ नंबर ऑफ क्रोमोसोम्स. अरे ovary is the 23 pair, but egg when it form ovary, it form the egg, its number of chromosome become half. Listen guys, which cell was used there? No, wool, no embryo. Actually, see, mammary gland cell was used. Mammary gland cell was used. Mammary gland cell was used. Mammary gland means breast cell guys. Breast cell is a somatic cell, a reproductive cell, somatic cell. So mammary gland cell was used for the formation of this sheep clones. That was the first mammal clones. Okay, it was the first mammal clones. You people remember. So the first mammal clone was developed through mammary gland. And one more question guys. The first clone those developed in India, it was a buffalo clone. Okay, the first clone that was developed in India, it was a buffalo clone. Okay, that buffalo clone was developed in NDRI, National Dairy Research Institute. National Dairy Research Institute, Karnal, Haryana. This is the place where India has developed the first clone, and it was a buffalo clone. Name was I think Garima. Okay, but my question is here, which body part was used in the case of first clone that's developed in India? It was a buffalo clone. The answer is a ear cell. Answer is a ear cell. Ear cell means this cell guys, ear cell. And ear cell is also what? It is a kind of somatic cell. So ultimately now it is proof, if I want to develop a clone, so definitely you people will give me what? You people will give me somatic cell. You people will give me somatic cell. Okay, you all people will give me somatic cell. Now you people have an entire concepts. Okay, now you people have an entire concepts and nobody can confuse. This concept will be very helpful to you guys. Even those people have not a science background. Okay, still I am very sure this type of information you read in the school level. But how this information is connecting in the application of cloning, DNA fingerprinting, I have corrected. Okay, now I am very sure guys, when at the time of discussion of cloning, Okay, I'll ask you guys, give me your cell. I want to develop your clone. I'm very sure as a common sense, you people will give me somatic cell. As a common sense, you people apply your common sense and you people will give me a somatic cell. Okay, you people will give me a somatic cell. Now come what are inside the chromosomes. Now the discussion is what are inside the chromosomes. So nucleus, within the nucleus have chromosomes. Chromosomes are made up of. So give a separate heading. What is inside the chromosomes? Chromosome wala part is over. What are inside the chromosomes? Okay, what are inside the chromosome? Chromosomes are made up of. Here I'll tell you according to some models. Nucleosome, right? Some information. Nucleosome and solenoid model. Nucleosome and solenoid model, right? Nucleosome and solenoid model provide the information. Stem cell is somatic cell or germinal cell? Stem cell is a somatic cell. Where is a stem cell? Somewhere in the embryo, somewhere in the adult cells. Here I'll tell you embryonic stem cell and adult stem cell. But stem cell is overall this is a somatic cell. Okay, Abhinandan, it will be more clear. Give me some time, bacha. Okay, at least 2 दिन तो दो ना biotech finish करने में. अभी तो basics ही चल रहे हैं ना. Today, tomorrow, at least 2 days. You people should give me. So chromosomes, according to nucleosome and solenoid model. According to nucleosome and solenoid model. Okay, chromosomes are made up of. According to nucleosome and solenoid model, chromosomes are made up of DNA and histone protein. Chromosomes are made up of DNA and histone protein. Chromosomes are made up of DNA and histone protein. So chromosomes are made up of DNA and histone protein. श्रेयस सारे किए धरा पे पानी फेर दिया. Literally you are literally मतलब बच्चा you whatever I have done ना everything expired. I just want everyone answers. Those just Shreyas told me. Shreyas told me like does only certain parts of body has a somatic cell? I am very sure my soldiers are ready to give this answer. My question is, as Shreyas asked me, does only certain part of the body have a somatic cell? सारी किधर है पानी फोड़ दिया. मैंने कहा only reproductive parts only in the one part, that is the testes and ovary, rest of all somatic cell. अब you are asking me totally opposite contrast feature, only few part of somatic cell in the body. Does only certain part of the body has a somatic cell? सारा बर्बाद कर दिया. What about other options? Other options. I have done my entire work, whatever I have done, everything expired because of this answer. No, no, no. It's totally opposite questions. It's totally opposite informations. Except reproductive cell, rest of all somatic cell. It means somatic cell everywhere. Only reproductive is one place. Apart from this, see, everybody right, everybody right. Shreyas bhai, either you came late here, okay, yeah, you are not listening me properly. Okay, so give your attention. I have spoken you 3 times this statement, and even example also have given you, and it is the proof because everybody has given me right answer here. I thought you as so everybody right, all except the reproductive. Okay, reproductive is only one. So ultimately somatic cell is maximum inside your body, not reproductive cell. Reproductive is only one part of your body. I joined late. That is the reason. That is the reason. So this is a problem. Okay. मुझे लगा था there is something with you. अब देखो everybody, Ananya, okay, Aishwarya, Arya, okay, Arya, everybody right, everybody right, everybody right. That sabab the person was even confused with the stem cell. The stem cell is also somatic cell. Stem cell also somatic cell. Okay, fine. Come. So, according to nucleosome and solenoid model, chromosomes are made up by DNA and histone protein. Now where you people have reached about the DNA. Histone protein is not required. So histone protein discussion we will not do. But DNA is important, so we will go into the study of DNA. But one thing, guys, with DNA one more name come, that is called RNA. And DNA and RNA are called as a nucleic acid. The better you give a heading, nucleic acid. Better you give a heading, nucleic acid. Nucleic acid. Nucleic acid. Nucleic acid also called as a nucleotide. So better you people also write one more term. It is a nucleotide. In biochemistry, nucleic acid is also nucleotide. Shreyas, come on time. Come on time. I start my class exactly at 7 o'clock. Okay, exactly 7 o'clock. Come here on time. So nucleic acid, it's in the biochemistry. It's also called as a nucleotide. Okay, nucleotide. So some factual information I want to give you in the beginning. Who discovered nucleic acid? So behind it, Friedrich Miescher. This man discovered nucleic acid in human pus cell. In human pus cell. So this man discovered nucleic acid in human pus cell. So this man discovered nucleic acid in human pus cell. The first time nucleic acid was discovered in human pus cell. The first time nucleic acid was discovered in human pus cell. So first time nucleic acid was discovered in human pus cell. So first time nucleic acid was discovered in. Please discuss about RNA and DNA vaccine. DNA vaccines bata dunga, don't worry. It's not a difficult task, it's very easy. I'll discuss with you DNA vaccines. So who discovered the nucleic acid? Friedrich Miescher discovered nucleic acid in human pus cell. You know, sometimes your skin have a pus formation, dead RBC, some bacteria, you know, pus formation, yellow color. That you people cut it and remove it. Okay, so in that pus cell, first time nucleic acid was discovered. Okay, first time nucleic acid was discovered. Hello. Fine, first time nucleic acid was discovered. Now, so nucleic acid also called as a nucleotide. Actually, nucleic acids are made up by nucleic acid. Yeah, nucleotide is consist of nitrogenous base. It consists of nitrogenous base plus sugar and plus phosphoric acid. Plus sugar and plus phosphoric acid, right? Nucleic acid consists of nitrogenous base plus sugar and plus phosphoric acid. So within the nucleic acid and nucleotide, what are present? Nitrogenous base are there, sugar and phosphoric acid there. How many types of nucleic acid? So I hope you people know very well. In the school time you read it. The one is the DNA and second is RNA. So those DNA available inside our chromosome, it is a kind of nucleic acid. So those DNA available in our chromosome, this is the kind of nucleic acid. Okay, this is the type of nucleic acid. This is the type of nucleic acid. Remember here, guys. This is the type of nucleic acid. Now come. Okay, what about the difference between the DNA and RNA on the basis of nitrogenous base, sugar and phosphoric acid? Listen. So one side write DNA and second side write DNA and see differences of nitrogenous base, sugar and phosphoric acid. So one side is DNA, write down. One side is DNA. Second you write RNA. Actually DNA is very, very important, guys. Without a study of DNA, I cannot reach. Without the study of DNA, I cannot reach on genes. And without the study of gene, I cannot describe you biotechnology. So I have a need of again 30-40 minutes more. But after it, you people will be very clear where is a gene. And why the study of genes are very, very important. At least say 6-7 questions suddenly you start to solve after 30-40 minutes. And you know, every year UPSC has a question from gene sequencing, gene sequencing, gene sequencing. Every year one question UPSC is asking from gene sequencing, yeah, DNA sequences. It means somehow DNA sequences and gene sequences are same. And how they are same, today I'll prove. So don't miss a class, guys, and be concentrate here. I've now come into the DNA and RNA. I know in the 9th standard, 9th standard I think. 8th, 9th standard DNA RNA differences are there. Again, I'll start the DNA RNA. First, you write down the name of DNA. Deoxyribonucleic acid. Deoxyribonucleic acid. Deoxyribonucleic acid. This is a DNA. What is the name of RNA? Already it is written there, a ribonucleic acid. Swikriti, both concept will come under the discussions. वैसे तो you are right, definitely you are right, those you mentioned, but you just wait, I will prove it in the case of cloning and test tube baby. Test tube baby is also my part of discussion, here I'll discuss. Okay, so name is the DNA, deoxyribonucleic acid, and RNA is what? Ribonucleic acid, those I underline. The name of nitrogenous base, the name of nitrogenous base, the name of nitrogenous base, those are present in DNA. What are these? See guys, adenine. Adenine means A. Guanine. Guanine means G. Cytosine. Cytosine means C. Thymine. Thymine means T. AGCT. So which nitrogenous base in DNA? Adenine, guanine, cytosine, thymine. What are inside the RNA? RNA have a adenine, RNA have a guanine, RNA have cytosine, but thymine absent here. In the place of thymine, uracil here. In the place of thymine, here RNA have a uracil. So one side you got some side you will got a similarity, AGC same. Some side you will get a difference, thymine and uracil. Thymine and uracil. Next one is sugar. Which sugar in the DNA, guys? So here answer is the deoxyribose sugar. It's a deoxyribose sugar. So DNA have a deoxyribose sugar. What about RNA? RNA have a ribose sugar. RNA have a ribose sugar. So DNA have a deoxyribose sugar and RNA have a ribose sugar. So DNA have a deoxyribose sugar and RNA have a ribose sugar. So DNA have a deoxyribose sugar and RNA have a ribose sugar. Remember it. Okay, another one guys. Phosphoric acid present here. Phosphoric acid present here. And even phosphoric acid also present here. So phosphoric acid present both two sides. So phosphoric acid present here, phosphoric acid present here. Right? Here again you will have a similarity. Phosphoric acid and phosphoric acid. I need some more place. So better I write here DNA and RNA again. DNA and RNA again. Some more features here I will write. DNA and RNA. DNA, mostly you will get it. It is a double helical. Yeah, double stranded. DNA is what? This is double stranded. RNA is what? RNA is a single stranded. So DNA double stranded, RNA is what? This is a single stranded. So DNA is a double stranded and RNA is a single stranded. So DNA 2 strands are there and RNA 1 strand is there. So DNA is a double stranded and RNA is a single stranded. Okay, I'll show you in the picture, guys. In the case of DNA, okay, adenine connect with the thymine and guanine connect with the cytosine. But in the case of RNA, thymine absent. So here what happened? A connect with U and G connect with C. Actually here I'll give you one statement. As DNA is a double stranded, rarely some examples of animals or plants, it is a single stranded. Mostly RNA is a single stranded, but rarely it is also double stranded in some of the organisms. Okay, so what is here, guys? DNA mostly double stranded, rarely single stranded. RNA mostly single stranded, rarely double stranded. So remember it, guys. So DNA mostly double stranded, rarely single stranded. RNA mostly single stranded, rarely double stranded. So this feature you people remember. Okay, so remember this feature, guys. Double stranded and single stranded. Okay, double stranded and single stranded. So double stranded and single stranded. Now question is just like Anil asked me a question. Anil has given the statements that the main difference is the what? Double standard DNA. No. The main difference between DNA and RNA. Main difference between the DNA and RNA is not double and single stranded. This is on the basis of sugar composition. Main difference, if anywhere anybody will ask you, examiner ask you, what is the main striking difference between DNA and RNA? So your answer will be one side have a deoxyribose sugar and second side have a ribose sugar. Okay, one have a deoxyribose sugar and second one have a ribose sugar. This one you people remember. Okay, so deoxyribose sugar and ribose sugar. Even whatever I have mentioned, everything I'll show you in the pharma picture. Okay, I'll show you in the pharma picture. So I have one more question that is very, very important. What about the location of DNA? So your next heading is the DNA. You give a separate heading DNA. You give a separate heading about DNA. And first of all, here I have a question. Okay, location of DNA, right? Second point, next point, location of DNA. Right, the next one, location of DNA. Location of DNA. So now question is where DNA present? Location of DNA. So where is the DNA inside your cell? Inside your body, where is the DNA? So within the nucleus, everybody know. Within the nucleus, everybody know. Within the nucleus have chromosome and within the chromosome have DNA. So of course nucleus have a DNA. So one place to everybody know, nucleus have a DNA, everybody right. And a part of this guys, two more places where the DNA present, you remember, the one is mitochondria. One is mitochondria. And one more is called chloroplast. One more is called as a chloroplast. Photosynthesis involved. So these are the three examples where DNA present inside a cell. But chloroplast not available in the human being. Chloroplast is a feature in the plant body. So if I ask you where is the DNA in the human being, so of course your answer will be nucleus and mitochondria. And it was a UPSC question, guys. It was a IS prelims question. UPSC has twice this question in your paper. But whenever this question came, it was before 2010. Okay, so before 2010, two times this question came, where is the DNA inside a cell? So you people be ready for the next time. It's a nucleus, mitochondria and chloroplast because this question repeated. So by chance, by mistake, UPSC is going to take any of the previous questions, and this question UPSC asked two times already in your paper. So location of DNA, you again repeat guys, main DNA inside your nucleus. Okay, but a part of nucleus guys, some other places, so only few amount of DNA also available in mitochondria and chloroplast. And see, mitochondria and chloroplast, this is the reason because they also have a DNA. They are called as a semi-autonomous body. They are also called as a semi-autonomous body. So if UPSC asks something where which cell organelle, mitochondria, chloroplast are the cell organelles. Which cell organelles are semi-autonomous body? The answer will be mitochondria and chloroplast. Answer will be mitochondria and chloroplast. Okay, answer will be mitochondria and chloroplast. You people remember. Answer will be mitochondria and chloroplast. Now come next. Okay, come next. And see, this mitochondrial DNA is very, very important. During the case of three parents baby, this discussion I will do, and you people don't be confused. And that time I'll tell you, during the basic discussion I told you mitochondria have also DNA, and here I describe you mitochondria have also DNA. Now come DNA structure. And DNA structure, here your heading will be what? Double helical model of DNA, right? Double helical model of DNA. Double helical model of DNA. Double helical model of DNA. Actually, DNA is a two helix structure. It's a helical structure with have a two strands. And see, this model was given by, I hope you people know very well, Watson and Crick. This man very popular, Watson and Crick. I think everybody know the name of this person. And for this contribution, this person got Nobel Prize. And Watson and Crick got Nobel Prize for this contribution. So Watson and Crick got Nobel Prize for his contribution. Contribution is double helical model of DNA. So for double helical model of DNA, Watson and Crick got Nobel Prize. Okay, Watson and Crick got Nobel Prize. Up now see guys, double helical. DNA is a two strand, double stranded. I hope you know. DNA is a double strand. Double strand means two strand. And it's a helical like. Helical like means like this. Screw, I hope you have seen. Screw. Rope you have seen. Rassi. As something like this. In this way guys. Helical structure. Screw, something like this. It's a helical structure. So that DNA, which is a helical structure, and two strand of DNA, I'm going to make here on the board guys. So how this entire structure I will make, but you people know very well, this board is the 2D. And those DNA structure I'm going to draw here, it's a 3D form. How a 3D form picture I'll show you on the board, you just see guys. In this case of helical structure. Okay, fine. You just see. Double strand. And see one thing more guys, DNA is an anti-parallel. Anti-parallel means, see here. Suppose it's a DNA strand and two strands are there. Both side the cap is on top, but no, one side cap will be on bottom. It show its anti-parallel nature. It show it as an anti-parallel nature. It show here as an anti-parallel nature. Okay everyone. So up and down. It show as an anti-parallel. So DNA is an anti-parallel in nature. And two strand here. See the one strand. See the one strand. Okay. See the one strand of DNA. Here I'm saying it's a 5 prime. So here I'm telling you it's a 3 prime. It's a 5 prime. It's a 3 prime. So it's a 5 prime and it's a 3 prime. You people remember. Lochan, I'll show you helical structure. First you see my helical structure, those I am drawing. DNA is a helical guys. So if here have a 3 prime, so here have a 5 prime. Actually 3 and 5 prime have some specific features. Phosphoryl group and hydroxyl group. But here in this GS class it is not required. Okay, you just remember something is 5 prime, something is 3 prime. And what is the parallel here guys? The second strand of DNA, those are here, see, those are the here guys. Parallelly the another strand is like this. Parallelly the another strand is like this. Parallelly the another strand is like this. Here I am telling you it's a 3 prime and opposite it is a 5 prime. So what it show guys? It show guys DNA is an anti-parallel. Means DNA is like this. DNA is like this. And but it is in the coil form. It is in the coil form like this. Okay, like this. DNA is an anti-parallel. Okay, DNA is anti-parallel. So I hope you people very clear what is the concept of anti-parallel, nature of DNA. Okay, and see one thing more guys, one thing more. Here this is DNA. Here this point, and this point is a one helix. Okay, one helix. So what you got here? It's a one helix. Either you call one helix or one coil. This is what? This is one more helix. Either you call one helix or coil. So it's a one helix, So one helix or one coil, you must be clear. So one helix or one coil, you must be clear. So one helix or one coil, you must be clear. One coil means how much it is 360 degree. One coil means how much it is a 360 degree. So 360 degree here, 360 degree here. One helix or one coil. Okay, one helix or one coil. Now come. In this one helix, here DNA is going to coil 360 degree. Here 360 degree. It's a very simple guys. If a DNA is here, and after coiling DNA is going to come here again. A, like this. One coiling happened. But when the one coiling happened, DNA reached here. When further one coiling happened, DNA reached here. So it's what? DNA is here. After one coiling, DNA reached here. So it's what? It's a 360 degree. But one helix or one coil. Further DNA after one coil reached here. One more helix. Okay, in this way have a DNA structure. And see guys, this distance is 34 angstrom. This distance is 34 angstrom. So this distance 34 angstrom, this distance 34 angstrom. I did not make it. Watson and Crick. That man already got a Nobel Prize. I do not want to further than Nobel Prize. It is proof. Whatever the proof I am telling you. And see in this DNA, you people know very well base pairs are there. What about the base? Base are ATGC and its pair ATGC base pairs. I told you guys DNA connect. A connect with the T and G connect with the C. A connect with the T and G connect with the C. Adenine, thymine, guanine, cytosine. So adenine, thymine, guanine, cytosine. You people remember. So adenine, thymine, guanine, cytosine, ATGC. Okay, adenine, thymine, guanine, cytosine, ATGC. So you people remember it. Okay, now you come here in this case. You just come here. In this helical structure, total 10 base pair here. Total 10 base pair. Base pair means AT is a base pair, GC is a base pair. Total 1 helix have a 10 base pair. It is very, very important, guys. If I will not discuss the base pair now, I cannot show you where is a gene. Because within this base pair have some specific character is a gene. Okay, so I cannot describe you what is the gene. Okay, and whenever I'll go into the gene editing, yeah, I will go into the gene therapy. So how I can describe you? So just give me 5 minutes more. Okay, now come here. About it. Total 10 base pair. 34 angstrom have how many base pair? 10 base pair. 34 angstrom how much base pair? It's a 10 base pair. The distance of one coil is a 34 angstrom. The distance of one coil is about 34 angstrom. And see the base pair. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 base pair. Okay, 10 base pair. And similarly again here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 base pair. And I will write some name of base pairs. That is very, very important. I will write down the name of base pair. See guys, name of base pair. A, T, G, C, G, C, C, G, C, G, T, A, C, G, G, C, A, T, T, A. Randomly I write about the base pairs. So total how many base pair here? 10 base pair. Similarly here in the next, guys, in the next, I can write any other like here AT start, here I said G, C, C, G, A, T, A, T, A, T, T, A, G, C, C, G, A, T, T, A. So don't think so the same repetition here and here. Any sequences are possible. In this 10 base pair, 10 base pair, further 10 base pair, further 10 base pair. It is just a small segment of DNA. You just draw this picture. You just draw this picture. This is important. You will just draw this picture. Okay, everyone. It's a helical structure. In that way, guys, have an entire DNA structure. Okay, and you all people have a very specific. Actually, you know, in the case of DNA fingerprinting, what we are going to match these sequences in throughout your body. Each and every cell have a same. Okay, throughout your body, each and every cell have a same DNA sequences. You people clear, guys? You people clear, guys? And this is the what we people match in the DNA fingerprinting. We people are matching what the DNA base pair sequences. Okay, we people match DNA base pair sequences. So you remember, guys. Okay, we people match DNA sequences. Now come. Okay, very advanced level of discussion, and here I will prove you about the human genome. Okay, here I will prove you about the genes, and here I will discuss many other questions. Those are important. See. I just erase it. And some more discussion, those are required. Give a heading, guys. Give a heading. Heading is here. Like replication. Replication. Then we'll go into the discussion of transcription. Transcription. Post-transcription. Post-transcription. translation, gene, and one more concept is the genome. Gene, and one more concept is called genome. Okay, one more concept that is called genome. So this is the entire heading, guys. Here, replication, transcription, post-transcription, translation, gene, and genome. Give heading. All entire discussion I'll do. Gene and genome. All entire discussion I'll do. So write it first. So write it first. Now come here. Some outline I'll give you that is very, very important. You come here in this outline. Okay, here I'm going to discuss with you about the human genome. Okay, what is a human genome? And here I can describe you guys. First of all, if I want to your gene, your genome, so definitely it is available inside your cell. It is available inside your cell. Better you people give me one cell first. Okay, give me your cell. Give me your cell. Fine. So it's your cell, guys. One cell. One cell. You people have given me one cell. What are inside the cell? What are inside the cell? Nucleus. So within the cell have a how many nucleus? One nucleus. I want everybody respond me, guys. Okay. Within the cell have a how many nucleus? One nucleus. If you people will give me a single cell, guys, you people will give me your cell. So within the cell I will get one nucleus. Within the cell I will get the one nucleus. Okay, one nucleus is here. Inside the nucleus, what are present? Within the nucleus have a presence of chromosome. Within the nucleus have a presence of chromosome. What about the number of chromosome? 23 pair chromosomes. 23 pair chromosomes. Okay, within the one nucleus have a 23 pair chromosome. See in this chromosome, 23 chromosome, your father chromosome. Another 23 chromosome, your mother chromosome. Actually, inside your each and every cell, those chromosomes you have, here 23 chromosome, your father chromosome, 23 chromosome, your mother chromosome. So 23 your father chromosome and 23 your mother chromosome. Okay, what the next? Within the chromosome, what are available? DNA available, DNA available, DNA available. Now my question is, in this 23 pair of chromosome, those available in one nucleus and one cell, how many DNAs are there? So you remember, one chromosome have one DNA. So total how many DNAs are there? 46 DNA. So again I can tell you 23 DNA you got from your papa and rest of the 23 DNA inside your cell you got from your mother. Your mother. And in this 46 DNA your entire genes are there. Okay, entire genes are there. This is called your genome is there. If this is your cell, so definitely it's your genome. That's why I told you, you people give me your cell. And how to describe the genome? Sum of all the gene. Sum of all genes. Sum of all genes, that is called genome. Write down first. Then I will involve into the proof of gene. Where is the gene? Sum of all genes, that is genome. Right. Sum of all genes, this is called genomes. So sum of all the gene, that is called genome. Write down. Now come here, guys. The question is about human genome. About the detail. What actually the human genome is. Try to understand here. I just want to write something about the human genome. Give a heading. Human genome. Human genome. How to describe human genome? Human genome. Generally, what is genome? So I told you sum of all the genes. What is the genome? I said sum of all the genes. Sum of all genes. Generally, it is the genome. So if I ask you what is a human genome, the total gene inside the human body is the human genome. Dog genome, total gene available into the dog, responsible for the dog formation, it's a dog genome. Total gene responsible for the formation of neem tree, that's a neem genome. Total gene responsible for the formation of banana plant, it's a banana genome. So generally sum of all the gene and number of genes change in everyone. Okay, अब your next question is, sir, tell me sum of all the genes. How many genes are in the human beings? Okay, earlier the gene that was discovered, I remember it was around 30,000 genes. Earlier 30,000 genes were discovered. Today it has reached around 42,000 genes. So total gene which are responsible for the formation of human being is this one right now. Because a lot of research going on. Okay, a lot of research is going on. 42,000 genes. Okay, fine. If you go into the detail of genome, the genome is called haploid set of chromosomes. Okay, haploid set of chromosome. Okay, haploid set of chromosome is if you have a 23 pair chromosome, so in 23 chromosome. Okay, because here if you people go into the dominance, alleles, lot of the concepts are there. But I'm not going to discuss into the allele level because I'm taking a class for just a UPSC. I'm not taking classes my campus for IIT Delhi, BTech, wala, Biotech, okay, MTech. Okay, now I'm not taking that classes. It's not required. But I don't like to go up to that level. Recipe, I'm not here to show my knowledge and everything. Okay, what I have done in the biotechnology and all. So you just see here, guys. What about the error, Muskan? You have a problem. Tell me. Okay, the sum of all the genes around it. If you go on the Google, maybe somehow the value you will get changed. But don't go on the facts. Try to understand the concepts. Okay, I just came here to brought all the concepts very easy for everyone. Okay. So, fine. Sum of all the genes around 42,000. Total these genes available in around 3 billion base pair sequences. 3 billion base pair sequences. So 3 billion base pair sequences. 3 billion base pair sequences. I think 3 billion, 3 into 10 to the power 8 or 3 to 10 to the power 9 base pairs. What okay, Muskan? Well, where error and now suddenly why everything okay? Fine. So around 3 billion base pairs. Okay, I'll discuss everything. What is the meaning of it? Okay, 3 billion base pairs. Okay, 3 billion base pairs. Okay, so sum of all the gene, it's around 42,000 gene, and this is available in 3 billion base pair. Okay, now come here, guys. Now come here. That is very, very important, and try to understand. Actually, all genes are available here on 3 billion base pair. Now you try to understand what is it. Suppose this is the nucleus. Suppose this is the nucleus. Suppose this is the nucleus. This is one chromosome or one DNA, second chromosome, second DNA, third chromosome, third DNA, fourth chromosome, 4 DNA, 5 chromosome, 5 DNA, 6 chromosome, 7. In that way, I have a 46 DNA. Suppose I just told you it's a 46 DNA. What are inside the DNA? What are inside the DNA? 1, 2, 3, 4, 5, 6 and base pairs. 1, 2, 3, 4, 5, 6 and base pair. 1, 2, 3, 4, 5 base pair, base pair, base pair. Why ATGC, CG, ATTA, ATTGC, जो those people have seen in the case of DNA structures. This is here. Okay, everyone, fine. If you count all the numbers of base pairs in one nucleus, where have a 46 DNA. When you count all the DNA numbers, within the DNA, those sequences of bases are there, you will get the number of base pair sequences of 3 billion. And this is the total number, okay, of base pair sequences, AT, GC, CG, AT. Means if you people count the AT, GC, base pair sequences, total in you all people, in your single nucleus have this much length of DNA. Everybody tell me you understood or not. Within your single nucleus have 46 DNA. Then the reference of base pair sequences, what about the length of all the DNA inside your single nucleus? So we all human being have a 3 billion base pair. If you are a human being, so definitely you will have a 3 billion base pair. अब 3 billion is a 3 to 10 power 8 or 3 to 10 power 9, you find, but 3 billion base pair in all of you. So 3 billion base pair in everyone, guys. Okay, so what is the human? Human is human because inside the human have a 3 billion base pair sequence of DNA. Okay, it is being added. If somewhere have some fossils, you don't know which organisms is there, but inside the nucleus and chromosomes and DNA, if you check and 3 billion base pair sequences you got, it's a human fossil. Somehow it will also help you to find out this answer. Okay, somehow you people will understand in this level. So 3 billion base pair, and see in this 3 billion base pair, total this much of gene available, those are discovered. Even if you people have an old NCRT, maybe 23,000, 24,000 genes are written. Today if you go on Google, let me check today what is the data. Today what is the data? What is the human genome size? Okay, total number of genes discovered just on Google. Definitely today we'll have some new values. Total number of gene in human body. You just listen what is the current value. It's a old data of estimated is 20,000, 25,000 gene is mentioning there. Do humans have 80,000 to 10,000? 1 lakh? Possibility there, guys. Everywhere have a different data. So don't be confused with the factual data. Okay, so here the data can be of just a minute, better I'll start from the 0 level. Here the data can be of 20,000 to 42,000 tak bhi data you can find out. So don't be confused here. Okay, don't be confused here. So 20,000 to it can go up to, you know, the huge numbers. But I'm not going there. Has raised total number of genes at least 46,831. Well, don't worry. Time to time new study. But yes, everybody should be clear. In this 3 billion base pair, total genes available. Those are responsible for the formation of you. These base pairs, everybody have a same 3 billion base pair. We all human being. Either you are a male or female does not matter. But inside the 3 billion base pair, this gene value varies. This gene varies. This is the your shape and my shape is different. Your color, my color is different. Your characteristics are my characteristics different. It is what we all people have a 3 billion base pair, but the number of genes it varies, and that's why you all people have a different faces, different characters. You all people have a different characters. You people remember. अरे ठीक है 3 बिलियन या 3.2 बिलियन, अब कौन है ये? शशि. स्टार्ट गूगल शुरू हो गया, सही हो गया. अब 3 बिलियन 3.2 ठीक है, चलो. मतलब यू पीपल नेवर फोकस द कंसेप्ट. आपको चाहिए ऐसा टीचर आ जाए जो 3,32,545 नहीं 547 वो वाला टीचर बताए जिसका कोई लेना देना एग्जाम में नहीं है. ओके, अब नाउ कम इवन यूपीएससी कैन नॉट आस्क यू द नंबर ऑफ जीन विन. क्यों? जब तक यूपीएससी विल मेक अ क्वेश्चन है तब तक 2-3 जीन विल चेंज. सम न्यूज. अरे सी 10 years back it was not discovered who is the gene which is responsible for the formation of retinoblastoma cancer. Okay, so 10 years back discovered. Few years back I got the Alzheimer gene discovered. So slowly and gradually new gene in this 3 billion base pair is going to discovered and the number is going to increase. This is my logic. Okay, so roughly you remember it's a 3 billion base pair. Okay, now come. Now the question is where are these 20,000 or 30,000 genes? Okay, in this 3 billion base pair sequences. And one more statement, those I want to sum up here, and then I'll go into the discussion of all replication transcription. You all people are human being, okay fine. You all people have a genomic size is how much? 3 billion base pair, okay fine. Up here in these 3 billion of base pairs, yeah, whatever this Shashi, who is telling? Shashi 3.2 roughly around billion base pair. Okay, what about the number of gene of Mr. अब जैसे मैंने बोला Shashi is here. मैंने बोला Shashi, you have a total 3.2 billion base pair sequences. Here the number of gene you have 22,234. Second guy Adarsh I'm telling you Adarsh, you have 3 billion base pair of sequences you have, but your number of gene you have a 32,224. And the next one is a Muskan is what? Muskan have a 3 billion base pair of DNA, but in this DNA have a 29,882 gene. Just like Akshay have a what? Again you have a 3 billion base pair sequences, but your number of gene inside your body is what? 37,224. I hope you got it, guys. So genome size everyone have a same, like 3 billion base pair or 3.2 billion base pair around, but inside it the number of gene vary in everyone. Tell me yes sir, I understood. Okay, yes sir, you people are human being because of this, But you people are different human beings because of this. Okay, and these all call as a human genome. In this way, human genome is defined. If you go on the advanced level, proper study molecular biology, to haploid set of chromosomes, alleles type of discussion required. But here you go according to UPSC, your answer is still correct, 100% correct. Nobody can complain you, your answer is wrong, 100% your answer is correct. Okay, 100% your answer is correct. And whatever the question will come, okay, you will solve it. Now last, sir, show me where is the gene inside this base pair sequences. Simple. So last you show it in this base pair of sequences, where is the gene? Definitely I will show you. It's a last discussion. Come here. It's a last discussion. Come here. I hope now you people never confuse. If you people will give me a single cell, so you are giving me your entire character, because you are totally here inside your cell. This is the reason, na? If I take your single cell, so ultimately you have given me all the genome, those are responsible for the formation of you. If I develop your single cell, ultimately it's you totally develop in the case of cloning. It means you totally develop in the case of cloning. Everybody clear? Okay, and you people very clear. If I, through the help of cell study, nucleus study, या chromosome study, या DNA study, ultimately here I will do the study of genes. So where is the gene? Gene available in DNA, या gene available in chromosome. I think nobody have any doubts. So gene available in DNA or gene available in chromosome. Anywhere UPSC asks the question, you people never confuse. Within the chromosome have DNA, within the DNA have a genome. UPSC can directly give you within the chromosome have gene, so it's right. But the proper order, within chromosome have DNA, within DNA have a gene. This is the correct order. Multiple times I am revising because biotechnology base at least 2 questions minimum, 3 questions, rarely 4 questions also possible, but 2 to 3 questions every year UPSC asked. And here question is totally conceptual. Okay, now come. Now we'll come into the discussion of replication. Now you just imagine, guys, first day when we all people form. When first day we all people form, it was a one cell structure. Okay, fine. And the name of cell was zygote. Name of cell was zygote. See here. First day you people were the zygote. Zygote means single cell structure. Zygote means one cell structure. So first day you people was one cell zygote. So first day you people was one cell, it was a zygote. One cell, it was a zygote. Okay. One cell zygote. What happened next? See guys. What happened next in this case? See guys. This zygote developed into the mother body. Okay, fine. Achha, in this zygote, how many DNA was there? I hope everybody know. 46 DNA was there. Now nobody have any doubt. Through the help of this flow chart, everybody clear. Okay, everyone? Fine. So it's a zygote 46 DNA. You know what happened, guys? What happened? This DNA, inside the mother body. When we people even develop. When we people develop this DNA inside the mother body, divide and redivide. And this DNA form multiple number of DNA. So DNA synthesize multiple number of DNA. Process I'll discuss with you. Then this DNA synthesize different number of RNA. Okay, see how you people develop inside the mother body. You got some DNA divide from more number of DNA, because in the beginning few DNA only, 46 DNA only inside your single zygote cell. Further the DNA divide, more DNA increased. Okay, then the more DNA synthesize more RNA. Then this RNA synthesize different types of RNA, like ribosomal RNA is called rRNA, messenger RNA is small mRNA, and transfer RNA is small tRNA. So ribosomal RNA, messenger RNA, and transfer RNA. These are different types of RNA. Ribosomal RNA, messenger RNA, and transfer RNA. These are different types of RNA. And see guys, these all the RNA. Ribosomal, messenger, and transfer RNA. They synthesize protein. And you must be clear, guys, your all the character inside your body, it is in the form of protein. So your all characters synthesized inside your body, it is in the form of protein. So your all characters synthesized inside your body in the form of protein. Now here I'll add on some informations. What about the information? See. DNA to DNA formation is replication. DNA to RNA formation is called transcription. RNA to various type of RNA formation, post transcription. And RNA, different types of RNA to protein formation, it is a translation, right? As a flow chart I mentioned. Right, as a flow chart I mentioned. It will take time. Actually, here I'll tell you, in this DNA have what? Genes. In this DNA have a genes. Suppose you have a 24,000 genes. In this DNA have a 24,000 genes. In this DNA have a 24,000 genes. Suppose you have a 24,000 genes, okay? And genes are responsible for the formation of characters. So I'm telling you, suppose 24,000 genes are responsible for the formation of your all proteins, your entire body. Your entire body base protein characters developed by 24,000 genes. Okay, so 24,000 genes, those are present inside your 46 DNA. It is responsible for the formation of your whole character. Okay, if I ask you guys, my eye color is red, so I have a red color of eye protein formation. Yeah, my color is black, so I have a black color eye protein character synthesized. And why I have a black color of eye protein synthesized? Because inside my body have a black color of eye gene. Very simple, guys. Suppose I am suffering with sickle cell anemia disease. So why I am suffering sickle cell anemia disease? Because here there is a one sickle cell anemia gene. And when this gene develop, it form a sickle cell anemia character. It form a sickle cell anemia character. And this is the reason when the sickle cell anemia protein synthesized, I am suffering with sickle cell anemia disease. I am suffering with sickle cell anemia disease. You must be clear, guys. I am suffering here sickle cell anemia disease. Okay, it means inside my body have a one gene. Those are responsible for the formation of sickle cell anemia in this all 24,000. And how the sickle cell anemia, even one day when people synthesize into the mother body, it was a cell, one cell. But when the baby developed as a 3 kilogram baby, and by birth I got sickle cell anemia protein, character. Because I have this defected gene, those I got from my parents. Either my mama or papa, they are suffering with this type of disease. They have this type of gene. And this is the reason whenever I form, through the help of the chromosomes, DNA, that gene come inside my body. And when I start to develop in 9 months or 6-7 days, finally as a 3 kilogram baby, when I form, this all the protein expressed inside my body. And those protein expression happen by birth, the same expression throughout my life. And I will suffer with the sickle cell anemia type of disease. Clear? How? Up see, guys, these all the characters, those I'm telling you, these all are the genetics. Generation to generation, generation to generation. And this is the reason genes are called as a hereditary characters. Heredity means those are coming from generation to generation. In the school time you're at it. Okay, and that's it. I hope you people know very well, we people got chromosomes from our parents. Within the chromosome, we got DNA from our parents. Within the DNA, we got gene from our parents. Now the question is, where is the gene inside the DNA? The last discussion. As here 46 DNA have a 24,000 gene. In this way have a various characters in inside your body. And those genetic character you people got. Now throughout the life you have it. As a very simple, my hair is not very straight, guys. See. Something straight, something curly. Okay, I think something straight, something curly. Thoda teda meda, not exactly straight. I'll go and do my setting, permanent setting, straightening. Okay, straightening. Mostly I've seen the girls are doing it. Okay, male guys में तो बाल ही इतना छोटा था, क्या straightening करेंगे? हमारा बाल तो ऐसे ही थोड़ा सीधा मेटा टेढ़ा बेड़ा रहता है. So I just ask you guys, in the case of straightening, okay, why I'll waste my money? Because I know very well inside my body have this type of genetic characters. Those are responsible for the formation of tedha meda gene, tedha meda hair formations. If I'll do the permanent setting against the new gene, against the new hair when synthesized, my hair is already of the curly type. So again the hair that will synthesize the new hair is a curly type. So permanent, nothing will be permanent because gene already have it. Even I'll do some discussion about the gene therapy. I'll do some discussion of gene editing. But it's a different part. First of all, you listen guys, though genetic character I got it. Now my hair cannot be permanent. Even see guys, see. My look is totally like to the South Indian types. Okay, this is the reason when I go to Bengaluru, Hyderabad, in these areas, I easily mixed up. Whereas people think, oh, same color of the person is coming. Okay, so my is a tall, dark, handsome. Don't mind, I'm telling you about me, myself. I'm handsome, so you decide, okay. So I'm just telling you about it, okay. Even those color I got it today. Why the color is this? Here the reason is my father. I don't like to say that because I cannot blame my father that because of my father I got this color. But I'm telling you guys, those color I got it. It's from genetic character. Okay, and that is already here. This type of color. If I do polish of my face, everything here. Okay, so what will be there? My face will become gura, white. No, never. Because my gene is already dark type. So whenever the gene will form the character of my skin proteins and all, so whatever the character is what dark. So this is the reason my color is what? It's a black. I hope you understood. Okay, fine. So why I'll waste my money? कि बैठा हूँ सलून में 1000 रुपए 2000 रुपए लगाए जा रहा हूँ. Better you should be satisfied as God has made you. You are very special. Okay, so I hope you understood. Those genetic character I got it. This genetic character will not change. Some procedure we can discuss in the biotechnology class like gene therapy and gene editing, even CRISPR-Cas9. You also know very well this is very, very popular. So in that way, okay, some discussion I'll do. So this is entire thing, and I hope you got through the help of very interesting example. Okay, so I just suggest to everyone, you should not waste your money, okay? And whatever the God even made you, you are already very special, because in the respect of God, you all people are very, very special. You should not complain to God. Fine. Whew. Yes. Ab you come last discussion. Where is the gene inside the DNA? Last. Where's the gene inside the DNA? So suppose this is DNA. Suppose this is the DNA. Ab see guys, 3 billion base pair is not possible to draw here. Manjunath, Michael Jackson face surgery. This is the reason Michael Jackson suffered a lot of problem in their own life. This man was never satisfied as God has given their own natural conditions. Okay, this man never satisfied with as the God has made him. He always blamed to the God and changed their own characters. People to people guys, somebody's whatever they are getting in their own life, but they are still frustrated. They never thanks to God. Oh God, thanks God, I am living because of you. Okay, but expectation. Somebody's have a lot of expectations, and that is the reason they are suffering a lot. When the condition is adverse, they suicide actually. The people, those are more expectation. The better you create expectation less, okay, and try to be happy. But then it is a human nature, okay. That human never satisfy. Whatever the human have, again the problem is this. Okay, now you come guys, it's a DNA base pair, okay, DNA base pair sequences. Suppose it's 3 billion base pair sequences, 5 prime, 3 prime, 3 prime, 5 prime. Up 3 billion base pair is not possible to draw here guys. 3 billion I can draw. 3-4 days I'll draw everything on the wall. I cannot draw. Okay, fine. 3 billion base pairs. And see the base pair 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Suppose base pairs. And dash 3 billion base pair. Roughly it's a 3 billion base pair. Okay, and just randomly I'm writing the name of base pair sequences. Suppose A, T, T, C, G, A, T, C, G, A, C, G, G, A, T, T, A, C. Something I have written as a randomly. Okay, randomly I just have written here something. Okay, and now what the next part here? See guys, what is the complementary? I hope you know very well. A complementary T, T complementary A, T complementary A, C complementary G, G complementary C, A complementary T, A complementary T, T complementary A, C complementary G, G complementary C, G complementary A, T, T, A, T, and C, G. It's a base pair sequences. Okay, it's a base pair sequences. Now you come. What will here guys? What will come here? See. Suppose this DNA base pair sequences. Suppose these base pair sequences. After replication, transcription, post transcription and translation. Ab you people know very well what is it. Replication, transcription, post transcription and translation have a formation of sickle cell anemia disease. Sickle cell anemia protein have a formation of sickle cell anemia protein. If after it have a formation of sickle cell anemia protein, it means you are suffering with the sickle cell anemia disease. Okay, sickle cell anemia protein synthesize. So sickle cell anemia protein means you have a sickle cell anemia disease. So sickle cell anemia protein means you have a sickle cell anemia disease. And now this is what? This is the set of DNA base pair which are responsible for the formation of sickle cell anemia protein. After replication, transcription, post transcription and translation, this is called as sickle cell anemia gene. This is called as sickle cell anemia gene. Similarly, other base pair sequences. After replication, after transcription, post transcription and translation, responsible for the formation of black color eye protein. So black color eye protein means this is the character. Inside your body, all characters synthesized in the form of protein. I told you, fine. If you have any disease, any characters, everything is what? In the form of protein. And this sequence, I'll tell you what. This is the black color eye gene. This is black color eye gene. This is sickle cell anemia gene. In this way, different characters form. And finally, slowly and gradually, scientists identify in that way 3 billion base pair, 3.2 billion base pair have around 24,000, 26,000 genes. Ab I want everyone answer. Yes, sir, I understood. Where is the gene? You just define it. And how to conclude it? Just a minute. Genes are what? A very specific DNA base pair sequences. It means if anyone have a black color eye, it means you have this base pair sequences somewhere present inside your DNA, and when this DNA will synthesize for the formation of eye color, so eye color becomes a black color. Okay, yeah, if anybody have a sickle cell anemia disease anywhere, any people in the worldwide, so they have this type of gene sequences. Gene means a specific DNA base pair order. Those are responsible for the formation of a specific type of protein. And in that way, guys, 3 billion base pair have around 24,000, 30,000, whatever you say, total identified. Okay, total identified. If you people have any doubt here, you can ask me, because now I describe you about the gene, because now I'll go into the application parts. Questions are coming from the application part. So questions are always coming from application parts. So any question you have, so ask me, because now I'll go into the application parts. So better now you come into the application part now. Those are the demand of UPSC. I'm going to erase. Akshay, gene can be changed. Okay, definitely gene can be changed, and there is a various reason to change the gene. Okay, Maybe cancer is what? It's a kind of genetic changes. Those happen inside your body. Maybe because of radiations. Maybe because of chemicals. Okay, many reasons are there in the surrounding for the genetic changes. Okay, some happen naturally itself because of because of artificial. Okay, and mutation also there. Mutation is a sudden changes in the genetic characters. Okay, just like what happened in Hiroshima and Nagasaki when atom bomb dropped. Suddenly the genetic characters of the people change and people become physically disabled. They have not taken time for 3 generations, 4 generations. Okay, they have to suddenly change the genetic characters. Like ecotypes. Ecotypes is in the ecology. It's a gene ecology. On the basis of genetic characteristics of the plant also change. Okay, change. So depends condition apply. And mutation to suddenly change the genetic characters. Just like cancer. Cancer is a part of mutation. All genes are working properly inside your body. Suddenly a gene become abnormal and responsible to create the cancer problem. Definitely. Sir, parents eye color black but son eye color green. So is there any reason? हाँ क्यों नहीं हो सकता? Definitely it is possible. It is according to Mendel's law. Maybe your papa color is what? Papa color is the black and the kid's color is the green. Possible. Why not possible? Here the reason may be your dada dadi and nana nani, your grandfather grandmother. Maybe your father have a black color expression wise, but the green color is hidden. I told you na, Mendel's law of dominance, but that is not required here. Okay, just like I can tell you guys, my height is good. Why my height is good? My father height is not this much of 6 feet, but my mother was even 5 something. Okay, girls and 5'5" is good, but 6 feet definitely it's not came from my papa and even not from my mama. So I'll tell you here my dadaji was responsible because my dadaji height was very good. It means inside my father, my dadi gene was express and my dada gene was hidden. But when I form, so ultimately my dadaji gene that was hidden inside my father, it expressed me. So it means my father is not this much of height. My father I think it's 5'8" something around. Yeah, shayad 5'9". I don't think so. But I got 6 feet. So this type of situation is there. It does not mean कि अगर पापा मम्मा गोरे गोरे आप काले हो गए तो you start to feel अरे पापा ऐसे कैसे हो गया. अरे don't worry यार. Your nani was dark. Yeah, your dadi या dada was dark. Anybody's was there. So because we all generation to generation the character is coming. Sometimes your parents is also telling the character of this is came from your grandfather. Okay, this character was in your grandfather. You never seen, but I know this character from your grandfather you got it. So in that way, genetic and very cute discussion even your family members are doing. In Bhopal, what happened in Bhopal? Bhopal gas tragedy, something like it. So I think you people clear. I can enter into the application parts. Now nobody can confuse you what is the gene. Okay, gene are the specific base pair sequences. Those are responsible for the formation of a specific character, and character synthesized in the form of protein. Everything clear? So your all the characters of my height, color, hair, eyes, everything, any disease. Okay, everything behind it what? Genes are responsible. And I got everything from my papa and mama. This is the reason guys, you all people look like your parents. You all people look like your parents. And we all brother and sisters are look like our parents. Yeah, we all people match. Why? Because we all people came same gene from our parents. At least 50%, 60% genes in all brother and sisters. Because they are the origin of same parents. Even if you people have a 3-4 brother sisters. So better you sit between your parents. And it's a very cute discussion people start. Hey papa, I got your eye color. If mama eye color I got, it's looking blue. You have a green, though I got green. Papa, it's nose something very different. It's because of your nose. If I got this one from the mother na, so it's very cute discussion people are doing. Why brother sister matching? Because definitely they are getting the same gene at least. So look wise you can find because same mother and father say they form. So ultimately genes are same. The characteristics are same. Very cute discussion. Okay, fine. Very cute discussions. Now comes some application part. This topic is very interesting, guys. We people can flow out of the syllabus. So better we stop and come on the point. Biotechnology made easy. Don't worry, I made every subject easy. Okay, whatever I discuss, don't worry. Now you come here. Into the application. Actually, you just come guys, you just come. How DNA studies are important for us? If I ask you a single question and you people start to give me answer. How DNA studies are important for us? How genomic studies are important for us. How DNA studies or chromosome studies are important for us. So I hope you people can say, sir, we can find out genetic defects. We can find out genetic characters. Those are coming from generation to generation. Through the help of these DNA studies, it is possible. Even through the help of DNA studies, we can find out parent-progeny relationship. Parent-progeny relationship. Parent-progeny relationship. I'm the son and daughter of this parent. You can check. Through the help of DNA study or genetic study, or chromosome studies, genetic defect we can check. We can do the genetic defect. We can check the genetic defects. Those are coming from generation to generation in some family members. And of course, guys, whenever we'll do the defects in all studies through the help of DNA analysis, so we can remove it also through the genetic engineering technology. And when the people remove this, when the people remove it, so finally what happened, guys? We can do the correction of genetic defects. Okay, we can do correction of genetic defects. So correction is possible. Correction is possible. Those defects happen, that correction will do. But for the correction, guys, what is the procedure for the corrections? That discussion I'll do into the applied areas of biotechnology. But first of all, detection is important. But definitely through the help of DNA studies, if you'll find out the genetic defect, so that genetic defects wala part, we will cut and remove outside and add on the normal sequences. How it will be there, it is a part of genetic engineering. That discussion I'll do in the biotechnology. But whenever we people can do the cut the defective gene from your body and integrate the normal gene sequences outside the body, the next generation is totally free from the genetic diseases. अब suppose mama have a sickle cell anemia, papa have a sickle cell anemia, and this type of baby 100% as a male or female who will born, they will 100% sickle cell anemia. So we can do something, genetic screening we can do. Papa side sperm, mama side egg. You take it through ejaculation. Papa and mama will give their own sperm and egg. And now what happened, guys? In this sperm and egg, you just screen where is a sickle cell anemia gene. Okay, and the gene already discovered. It's a gene sequences, those are responsible for the formation of sickle cell anemia. Father side in the sperm, that gene remove. Mother side egg, that gene remove. And now the normal sequence will add in the sperm and in the egg. And now the sperm and egg have a what? There is a treatment of the sickle cell anemia. Now this sperm and egg have no genes of sickle cell anemia. It will fuse outside the mother body, zygote form. And now this zygote after some division implant into the mother body. And now here those lady will develop a baby. That baby will free from the sickle cell anemia. But the sickle cell anemia knowledge you got through the help of DNA frequencies. CRISPR वगैरह is very advanced technology. Abhi you just come into the basic parts. Suppose there are some areas in the agriculture field. In agriculture field, I can see, guys, because of drought, because of flood, because of some attack of locust, insects, some attacks. Okay, lot of the plant die. I'm telling you, in these circumstances, drought, flood, many reasons, lot of the plant die, but still some plants survive in that locations. So you just do one study, guys. You just check which DNA present here inside. Those are responsible for the resistance of this plant, guys. That's why some of the plants survive. Those are responsible for it. Okay, so you must have to know there, guys. Okay, what will come here? Listen. You just check which genes are responsible to make a resistance of this plant. That gene you identify into the plant. That's why the plant safe. And transfer into all those plant die because of drought, flood. Different condition have a different gene. Attack of locust. And finally what happened? We can save the plants against these type of diseases. And how it become possible? Through the help of genetic studies, DNA analysis. So we can make a plant as a disease resistance. We can make a plant against the drought resistance. We can make a plant against the flood resistance. This type of characteristics are possible. I hope now you people understood. Okay, why DNA studies, genetic studies are important. Actually, I could not enter into the subject wala part. I discuss only the basic part, but still you are able to solve some questions right now. UPSC has the basics. But what is going to be happen in the institutions? Institution is start from this now. And this part is totally missing. This part is totally missing. And that is the reason a student got a difficulty to understand what is the DNA, what is the chromosomes. Even student also not clear that chromosome have one DNA. If I call you that genes are in chromosomes, nobody have a doubt. Genes are the DNA, nobody have a doubt. Because within the chromosome have DNA, within the DNA have a genes. And what is the human genome? All genes, some, those are responsible for the formation of entire human. It's a human genome. A BT cotton, etc. So you will get it. Okay, don't worry. My motive never come directly jump on the subject. Okay, I want to discuss first the basic, guys. Now I can go and discuss the biotechnology properly, advanced level of discussions, and nobody have a problem in this understanding. Now everybody know why mitochondrial gene therapies are important. Reason mitochondria have also genes, and if this gene is defective, so definitely that therapy is important. And this discussion I'll do there. Where is the DNA? Of course, if mitochondria have a DNA and DNA have a gene, so some gene of the human being also available in the mitochondria. Common sense. Common sense. Okay, common sense. I just want to ask you one question of 2011. Okay, it was very good question. I'm very sure you people will give me a satisfactory answer. This question is actually page number 24.47. Okay, this is mentioned at page number 24.47. 24.47 and question number is 16. Question number is 16. Okay, let me discuss this question here. That is very, very important. Listen, guys. At present, question number 16, page number 24.47. At present, scientists can determine all statements are correct in application question are bad in UPSC questions. It is not so. It is not so. At present, scientists can determine the arrangement or relative position of genes or DNA sequences on a chromosome. At present, scientists can determine the arrangement or relative position of genes or DNA sequences on a chromosome. How does this knowledge benefit to us? Your examiner is really so nice, so sweet. Scientists determine the arrangement or relative position of genes or DNA sequences on a chromosome. Within the chromosome, those DNA sequences present here have a presence of gene. Examiner is written there. Only concept wise you can understand. Now the people will have a difficulty to understand the question also. And how this study of chromosome, those have a DNA and gene are important for us. How this is important for us? Okay. 1. It is possible to know the pedigree of livestock. 1. It is possible to know the pedigree of livestock. Okay. 2nd. It is possible to understand the causes of all human diseases. Okay. All human diseases. And 3rd, it is possible to develop a disease resistant animal breed. Which statement correct? 1 and 2, only 2, 1 and 3, all of these. I think so, guys, you will not turn your page, neither answer is already given there. Okay, you don't turn your page because answer is already in the book. Okay, because next time those questions will come, it will be not same, but something connected with it. If you will understand why it is so, so you can solve your questions. Okay, and why I have done this much of work in front of you, now you can understand. Okay, so 1 and 3 is right, are right. It is possible to know the pedigree of livestock. Yeah, definitely through the help of chromosome or DNA or genetic sequences, pedigree analysis is possible. What is pedigree analysis? Parent to progeny relationship. Parent to progeny relationship, next generation, next to next generation, this study is possible. 1. So it is possible to know the pedigree of livestock. 1. Right. It is possible to understand the causes of all human diseases. See, guys, common sense. All human diseases you cannot detect. Tell me, how many of you people can detect that next day you will suffer with the dengue? Acquired diseases you cannot tell me. Those you will acquire. But genetic diseases you can tell. But here UPSC asks the question, all human diseases? No, all not. Only genetic we can detect through the help of DNA studies. I hope you got it, the second statement, why it is wrong. Because all human diseases are mentioned and all are not right. Okay, and third one, guys, it is possible to develop disease resistance. Yeah, disease resistance, flood resistance, drought resistance. Okay, this type of condition we can develop here. This type of condition we can develop here. Actually, one more discussion I want to do here, that is called silent gene concepts. And it was a question I think in 2018, 2019. Listen, this time, okay, this time, okay, this time at the same time, I will discuss with you about the RNAi. 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 Because at this time you people can understand entire concepts. This time you people can understand all the concepts. Ranjan. Sir, when you say human have a 30,000 genes, it means in human there are 30,000 unique genes. As number of cells are many more. Sir, number of cells are many more. Mean in the billions. Hence a large number of genes are there. Arey, nahi Ranjan, you confuse. Those genes available in cell number 1 na, the same gene available in cell number 2. And the reason I told you because of mitosis. Why you confuse, bacha? Why you confuse, bacha? Why you confuse? Even those questions, Ranjan, as I think this doubt not any other people have. Those sequence in your one cell have the same sequence entire your each and every cell have. One cell have a one type of gene, second cell have a different type of gene, third cell have a different type gene. How it is possible? Throughout your entire each and every cell have a same DNA, the same genes, same genomes. This is the reason if any of the cell I will take, entirely I will develop you. Okay, I think everybody clear. Now come RNA. RNA is called as a RNA interference. RNA is called as a RNA interference. RNA interference. Okay. One more concept in the case of RNAi, that is called silent gene. This is called as a silent gene. Next one, guys, about PTGS. Many terms I am writing. Post transcriptional gene silencing. Post transcriptional gene silencing. PTGS, post transcriptional gene silencing. Post transcriptional gene silencing. Okay, post transcriptional gene silencing. So these discussion I'll do, but these all the discussions are connecting with this topic. This is the reason just after this topic I want to discuss it. Even biotechnology introduction now later on I will start, but at the same moment of time RNAi I want to discuss. Small break. Okay, 5 minutes. You take your break. So welcome back. And now you come into the discussion of RNAi. It's another part, and it is an important one. Okay, RNA interference, because RNA interference have a good future. Okay, RNA interference have a good future. What is the RNA interference? You come here, guys. You come here. As in this discussion of how DNA synthesize the protein, you people know very well. Okay, see, as how DNA synthesize the protein, everybody know very well. And here the process was, I hope you remember, the process was replication, transcription, post-transcription and translation, then have a character formations. Okay, then have a character formation. You tell me, guys, you tell me. Here, this is the gene. Suppose this is the gene, and this is responsible for the formation of, suppose sickle cell anemia. Those diseases I mentioned there. This is responsible for the formation of sickle cell anemia. Responsible for the formation of sickle cell anemia. Okay, it is responsible for the formation of sickle cell anemia. Okay, everyone. My question is here, those sickle cell anemia characters of protein is going to form here after replication, transcription, post-transcription and translation. You people know very well. Okay. My statement is here in between. Okay, in between. If I do this process, this mechanism stop. I stop the post-transcription process. So those genes, sickle cell anemia is going to express here. This expression will stop. I hope you are getting my point. I'm just telling you, if this post-transcription mechanism from RNA to different types of RNA formation, through the help of technology, I stop. Through the help of technology, genetic engineering technology, if I stop this procedure, so those RNA synthesize here, further the types of RNA and protein synthesis not happen. So those genetic expression was going to form in the form of sickle cell anemia. Now I can stop. You tell me, you guys clear or not? It means those gene expression was going to be happen inside your body. Through the help of genetic engineering technology, I made it silent. Its expression is not going to be happen. Okay, and why the silent? I make this gene as a silent. The reason is because of post-transcriptional gene silencing I have done. During the process of post-transcription, I stop this entire procedure. This is called post-transcription gene silencing. I hope you are getting it. And finally, though genetic expression was an adverse genetic character. Okay, we have to stop, and in that way we can also do the changes in the human characters and we can improve the characters. I want everyone answer, guys. Okay, I just want it, guys. Okay, I'll give you many example, Piyush, you just wait. First you tell me PTGS you understood or not, that terminology I just have mentioned here. PTGS is what? Post-transcription gene silencing. So those gene silencing mechanism I am doing here, it is at a level of post-transcription. Okay, it is at a level of post-transcription. Okay, fine. Natasha, good. Okay, Mahesh, good. Trisha, Niranjana, Shahana, everybody. Only girls are here. Where's the male guys? Arya, okay. Fine, clear, great. So, you people remember, guys. Okay, this is important here. Manav, Sunny, good. So, I hope you understood, guys. Through the help of silent gene, we can do lot of changes. Okay, we can do lot of changes in the genetic engineering technology. So, somehow, it is also the mode of genetic engineering process. So what is the silent gene? I hope you understood. The genetic expression is going to be happen. We stop that genetic expression inside your body. So that genetic expression will stop inside your body finally. And this type of gene become as a silent gene. How you can make a gene as a silent gene? Then the answer is at PTGS level, scientists achieved it. PTGS means whenever the post-transcription process is going to be happen, especially for the formation of any proteins or characters, we stopped the post-transcriptional process. And when you stop the post-transcriptional process, the other informations will not form. Other characters will not form. So here those silent gene character you people got, it is because of PTGS. Okay, PTGS. So I hope you people very clear. And finally what we have done, we have interfered the RNA mechanism. This is what actually, sir, here what you will call, sir, here RNA mechanism we have interfere. So RNA interference. And why interfere? Because we people stop the post-transcription mechanism. So RNA to next formation, RNA mechanism interfere here. So what is the RNA interference? I hope you understood. This mechanism is stop. Why this mechanism is stop? Because post-transcriptional mechanism we have a stop, and because of the gene expression was going to form, it become as a silent. So it is called as a silent gene. it become here as a silent. So it become as a silent gene here. Okay, it become here as a silent gene. Now I'll come into the application of silent genes. Application of silent gene, and many application I have given here, guys, and directly I will bring even suppose some of the hormonal defects are going to be happen inside your body. Okay, some hormonal defects happen inside your body, some cancerous problem is going to be happen inside your body. Because of this level of characteristics, we can stop the problems, and person who is suffering with the cancer can treat. Any person who is going to have any hormonal disbalance is going to be happen because of genetic influences. This type of characteristics we can solve. Okay, and many different types of plants even develop, and some example I'll give you. Many different types of plants even develop, and some examples you just listen here. Okay, some examples you just listen here. Just a minute. Some RNAi example I'll give you. Okay, and this question UPSC asked in 2019, guys. Okay, silent gene characteristics. It's mentioned on page number 24.21. Okay, the concept is given at page number 24.21. Page number 24.21. Page number 24.21, and some example of the use of post-transcription gene silencing. Okay, post-transcription gene silencing in the mechanism of crops, productivity of crops. Here we stop with the blue rose. The one example is a blue rose. Red rose are very common, but here I'll discuss with you about the blue rose. One more example I'll tell you: edible cotton. Edible cotton. Edible cotton. Tearless onion. Tearless onion. Okay, tearless onion. Even later on you should write decaffeinated coffee. Decaffeinated coffee. Decaffeinated coffee. Opium or poppy. So decaffeinated coffee, opium or poppy. And one more that is called flower sour tomato. The one more is called that is a flower sour tomato. These few application I want to discuss with you. This few application I want to discuss with you. Okay, what is the question, Krishma? So that silent gene is still there or it's taken out? Silent gene is still there. Now it becomes silent. Gene, those are responsible for some characteristics become silent inside your body. So silent gene is already inside your body. But silent gene you are going to make artificially, na? Ab nature में blue rose not exist, but we can make it through the help of silent gene. Cotton is not edible, but through the help of silent gene we can make it edible. Tearless onion, those onion when you cut have a tear, you can stop this tearless property not there. Coffee, those have a lot of caffeine, we can reduce the amount of caffeine there. Opium and poppy, that is not can be used as a vegetable, it's a narcotics, but we can use it as a narcotics. Flash have a tomato that ripening process was very fast, that is the reason lot of the tomato destroy, we can slow down the ripening process. These all the characteristics of the plants even develop through the help of RNAi, silent gene and PTGS. Ab how it is so, I will prove. अरे बच्चा जब PTGS ही eager you people have silent, तो how the translation happen? Translation already stop. Not have a common sense. Okay, if you are going on the path, in between you stop, so how the next path? It's already stop automatically. Protein formation not happen. Common sense. Okay, common sense. Okay, Piyush, it's a common sense. All three RNAs are silent. What three RNAs are silent? Acha, okay, understood. Actually, silent means RNA is being silent. Now the further process not going to be happen. Post transcription maximum not happen. Okay, because RNA stop to do work. Yeah, the RNA stop to do work here. Now here the work is stopped. So this process not happen. All this process not happen. All this process stop. So further the character those who are going to form here, this character not synthesized, finish. That character not synthesized. Silence gene in the parent body can be expressed in the progeny. No. No. Only in the case of you it happened. Okay. If up here there is a two questions as a two statement. As a Trisha asked me, there are two types of gene therapies there. If genetic gene therapy we have done germline gene therapy, so it will be silent for the next generations. If somatic cell therapy we will do, so it will not go for the next generation, only up to you. And this is the part of gene therapy. Those you are asking me that gene silencing go to the next generation or not. Here it is depend on the we people are doing the genetic silencing characteristics of gene therapy at germ level. Yeah, it's a somatic level. For this answer, for that purpose you must have to wait. Okay, you must have to wait. Trisha, okay, fine. Because there is two type of gene therapy is possible. The one is a germline therapy and second is a somatic therapy. Wait. Acha, you come here. I just want to finish this topic as soon as possible now. It's already stretching more and more time. What is a blue rose? Actually, you know guys, normal roses are red. Why the red rose is there? Because within the red rose have a gene. Those are responsible for the formation of this enzyme. So red rose have a gene. Those genes are responsible for the formation of this enzyme. Again I repeat, red rose have a gene. Those genes are responsible for the formation of this enzyme. But what will be here guys? What will be here? Actually in this case, what will do? Will stop the formation of this process. If we will stop the formation of this gene characteristics, so finally the color of rose will become shift from the red to blue. So very simple, why a red rose will become blue? Because red rose is red due to presence of dihydroflavonol reductase enzyme. And for the formation of dihydroflavonol reductase enzyme, inside a rose have a dihydroflavonol reductase gene. If the gene mechanism we stop, make it silent gene, okay, PTGS process, so finally dihydroflavonol reductase formation not happen and the color of the rose flower, it will turn from the red to blue. This is the characteristics here. This is the characteristics here. Okay, this is the characteristics here. I hope you people understood how it is so. How it is so. Hari Prasad, you are asking me about polygraphy. Yeah, pleiotrophic genes. If more than one gene sickle cell anemia, to stop this character, we have to stall all those genes in the different cells. You people are getting confused. You people are getting confused. And it's a big level of confusion though this started. Reason I could not discuss gene therapy, I could not discuss the gene editing. But you people directly enter there. I am just telling you some example of RNAi. Okay, where we are doing this activity. It does not mean दुनिया के सारे example इसी से होंगे. तो why you develop CRISPR-Cas9? Why you are doing the germline and somatic gene therapy? You just keep calm and those I am telling you just listen me. Neither I cannot take my class. It's very difficult to finish it. Okay, when the all biotechnology will over, now then you will understand which type of technology where is the good. Where the sui will work, na, in that place talwar not work, sword not work. Everyone have a different application in different places. Okay, so you just keep calm. Please keep calm. Okay, I just said DFR is what? It is one of the enzyme. Okay, that synthesized into the red rose. Why this enzyme synthesized? Because red rose have this gene. Okay, but what scientists have done? Scientists stopped the mechanism of this gene because of silent activities. Now this enzyme formation is stopped and automatically the red rose turn become as a blue rose. Okay, now this red rose become as a turn as a blue rose. This is the point. What happened the edible cotton? Very simple guys. Gossypol is the compound. Gossypol is the compound. Okay, gossypol is the compound and it is poisonous. Gossypol is a compound and it is a poisonous. Okay, and that's why the cotton seed is not edible. But through the help of RNAi mechanism, scientists stop the formation of gossypol and now this cotton seed can be edible. TLS onion. Why TLS onion is there? Actually, what about the chemical compounds? Those are available in the onion, and when you cut the onion, it will enter inside your eye and you have a tearing. So that name is called allyl disulfide. This is called allyl disulfide. Allyl disulfide. Okay, allyl disulfide. Okay, allyl disulfide. Okay, allyl disulfide. What will come here in the case of allyl disulfide? Better what you do? The allyl disulfide formation stop into the onion, guys. So those tearless characteristics are coming in the case of onion. Now it will stop and the onion becomes a tearless onion. And now this onion will become as a what? Tearless onion. Remember, guys. Now this onion will become as a tearless onion. So I hope people will clear here in the case of tearless onion. So why have a tear when you cut the onion, guys? The reason is allyl disulfide is there. Okay, allyl disulfide is there. So allyl disulfide formation is stopped through the help of PTGS mechanism, and now this onion will not have a tear. It will become as a tearless onion. Now come decaffeinated coffee. I have a question, guys. Why you people like coffee? Okay, why you people like coffee? So don't tell me it provides you the lot of energy. You are wrong. The main purpose is taste. But what is the negative in the case of coffee, guys? Negative in the coffee is what, guys? Okay, negative in the coffee is what? There is a formation of theobromine synthase. Theobromine synthase. Okay, theobromine synthase. Okay, and some technical name I'm writing here. N-methyl. You just listen. N-methyl transferase enzyme. Even this type of name never come in your paper. But I'm taking the classes, so it is must that I should inform you. N-methyl transferase enzyme. In a very simple manner, guys, why you people like the coffee? It's because of taste. Okay, taste. But what is the negative with the coffee? Caffeine. And caffeine is dangerous to you. If more consumption of caffeine you do, a lot of problems are possible. Because of heavier caffeine amount, EDH level down. Maximum urination happen. Chances of diabetes insipidus. I'm not going there, but I'm telling you guys, the more amount of coffee, it's not good to you. It's not good for your health. And the reason is because caffeine is there. Okay. I'm giving you one more options. And now the scientist has developed artificially through the help of genetic engineering technology. Actually, those caffeine formation happen into the coffee. It is because of N-methyl transferase enzyme. Okay, so those caffeine formation happen. It is because of N-methyl transferase enzyme. Okay, so for the formation of N-methyl transferase enzyme, have a gene into the coffee plants, into the Theobroma coffee or something else. Okay, they have a gene into the coffee plant. Those are responsible for the formation of N-methyl transferase enzyme. Okay, and because of this enzyme, there is a formation of theobromine. Theobromine formation happen. It's a coffee compositions. Theobromine formation happen. So what the scientists have done through the help of post-transcription gene silencing. Okay, this gene silent. This formation stop. Theobromine synthesis reduces. The amount of caffeine and theobromine reduces, and finally this caffeine will be not dangerous for your health. Okay, so this is the name, but I don't think so UPSC ask you the name. Okay, UPSC ask you the name. Anil Yadav, coffee provide a high energy too. I agreed. Okay, but coffee also provide you theobromine. That is not safe for your health. That is the reason through the help of post genetic silencing. Okay, post transcription gene silencing a silent gene. We stop the gene mechanism. Those are responsible for the formation of N-methyl transferase enzyme, and because of this theobromine synthesis level decreases, and theobromine is dangerous for your health. So finally this level decreases and you people can consume this coffee right now. Okay, I think I describe you very easily, guys. Next, two more example here. The one is the opium poppy. Opium poppy. Okay, opium poppy. Opium poppy is what? I hope you know posta. Posta. Okay, Papaver somniferum. Okay, a lot of plants like morphine, codeine, cinegreen, lot of the alkaloids develop from these plants. And this is the reason these leaves and all body parts, okay, not useful for the human being as a edible crop. But what the scientists have done through the help of PTGS mechanism, those all the alkaloids name I was telling you, alkaloids name those I was telling you. Okay, formation stop, and now this leaf can be used as a vegetable. So all the narcotics alkaloid synthesis stop, no formation here. Okay, the finally it is edible. What is a flax-seed tomato? This is again a very important flax-seed tomato. Okay, flax-seed tomato. Actually, you know, I told you it was a first genetically modified plants. Those got license for the human consumption. And the company those developed the flax-seed tomato, it was named as the Calgene. So Calgene was a company, I think Calgene was a Californian company. Okay, developed first genetically modified plants. Okay, in the lab, and the name was flax-seed tomato. It got license for the human consumption. This is a very good question. It's a very important question you people should remember. Which one was the first genetically modified plant got license for the human consumption? Flax-seed tomato. What was the beauty of this plant? Actually, tomato has a lot of issues. Whenever you people go in the market and purchase a tomato, you should prefer tomato should be something hard, not soft, because the tomato have a fastest ripening process, and that is the reason when the farmer is going to harvest the tomato from the field areas and send it to the market, guys, lot of the tomato even destroy in between because of fastest ripening process. Again I repeat, because of fastest ripening process. So what is here in this case? What is here in this case? Actually here in this case what we people are doing, see, fastest ripening process. In this case of flax-seed tomato, okay, we reduce the actually through the help of post transcription gene silencing, we reduce the softening process. And now the process of softening means the those ripening process was fastest, guys, it becomes slow down, and now the lot of tomato conserve. So ultimately the purpose was what? Reduce the ripening process. The purpose was to reduce the ripening process. Only application is important to reduce the ripening process, and that's why the lot of tomato is conserved and even safe. Okay, lot of the even tomato can be safe, conserved. Okay, safe or conserved. Okay, now come into the opium wala Yashaswini just told you, like opium have a narcotics. It's a drugs type of things. Okay, because of this type of chemical formation happen. That is the reason opium leaf cannot be used as a edible purposes or any other purposes. What scientists have done through the help of post-transcription gene silencing, they stop the formation of these narcotic formations, and now this leaf can be used as a vegetable. In a very simple way I define you. Okay, many other cancer therapy, hormonal deficiency in the human being. Okay, here post-transcriptional mechanism can also be useful in the coming future. So lot of the studies are going on in the PTGS. Okay. So I hope you people understood why at the same time in the beginning I just describe everything. Okay, I just describe everything. Come next. Actually those biotechnology discussion I am doing here. This biotechnology, everybody know this is the technology where living organism used. A technology based on living beings. Biotechnology, a technology based on living beings. Okay, everyone. This biotechnology, those are based on living beings. It is an interdisciplinary subject, and this subject is connecting with the many subjects. What are the subjects? Like biochemistry, microbiology, okay, biochemistry, microbiology, bioinformatics, okay, bioinformatics. So there are various subjects. With that subject, this subject is a interdisciplinary. So this is interdisciplinary with the various subjects: biochemistry, microbiology, and bioinformatics. And here in this beginning, you must be clear, guys. Biochemistry means living organisms, chemistry, compounds like proteins, fats, carbohydrates, even DNA, RNA, they all are the chemical compounds. They all are the chemical compounds. Microbiology, study of small size of organisms. Study of small size of organisms. Bioinformatics, still very, very important. Even a lot of the bacteria, viruses, okay, microorganisms, we are using into the various technologies. Okay, we are using to various technology. Another one, guys. Bioinformatics, living organisms, characteristics. In the case of computer processing, computer modeling, okay, we are providing all the information. Even in a very simple way, guys, all those research and experiments is going to be happen in biotechnology. All those research and experiments going to be happen in biotechnology. Every research details we are putting here under the bioinformatics centers, under the computer modelings, computer simulations. Just like human genome project data is safe. If you want to do any experiment on the human genome project, if you want to do any studies of the human genome project, so why you are wasting your time? That experiment already happened. This data is available on the bioinformatics center. You can go and take that data for your study purposes, for your study purposes, yeah, other purposes. Neither those work already happen. You are wasting your time. Every country have a bioinformatics centers. Even many years back, I think 7-8 years back, 10 years back, bioinformatics was a mains question paper. All studies of biotechnology, biochemistry, microbiology, all these data we are saving here under the bioinformatics. Okay, everyone. So you must be clear. Bioinformatics. Okay, transcriptome type of question UPSC has, I think 5-6 years back. All the genes, those are responsible for the transcriptions. Okay, all the genes, those are responsible for the transcriptions. That all the genetic study was a transcriptome. Okay, but in that time it was a current affairs in the news, guys, transcriptome. But what is here, I hope you clear, biotechnology is interdisciplinary with the biochemistry, microbiology and bioinformatics. Even if you people have done any degree courses, either in biotechnology or biochemistry or microbiology, 70% syllabus are same. 70% syllabus are same. Okay, 70% syllabus are same. Now this biotechnology, in the beginning I'll tell you, on the basis of applications, biotechnology is defined in various colors. On the basis of applications, biotechnology is defined in different colors. So on the basis of applications, biotechnology is defined in various colors. What are these various colors? See. Arey bacha, Arya, abhi why you are going for the apex body of genetic modifications? Okay. Either you people are checking my knowledge. I should have to discuss genetic engineering approval committee. And that is set up under environmental protection in 1986. Abhi, this is not required. You wait. I think sometime I realize now somebody's have no patience. Yeah, somebody's even check my knowledge. Don't worry, whatever you people will ask now, I know very well. It's my subject. Okay, and I spent my 20 years. It's not less. So you just wait. Abhi, this time I'm discussing whatever you people focused here. Okay, who is the apex body? Genetic engineering appraisal committee started to ask it. It's set up under environmental protection in 1986. Even GAC came 2 times. Okay, one time as an independent question, second time as a EPA wala questions. So how I can miss it? Wait, keep patience, guys. I want literally a student those have a patience. This is an exam of patience. Don't be hurry and don't jump. I just start the biotechnology. Definitely it will come. If I said the biotechnology over, and suppose that time I could not discuss, in that day you ask. Abhi just to beginning of it's just a 1.5 day. Yeah, half lecture I discussed last day. Today I'm taking the second day. Okay, so wait please. I think so you people have a doubt with me. Okay, I think so. And you people have a doubt on me. Okay. And sometime I think so you people check my knowledge. Okay, I give you the answer, still you people have not believe on me. You wait guys. 20 years I did not waste my time with you. Okay, many toppers, many failures. And there are around 1,000 plus students, they got success in UPSC, civil services. Proper IAS, IPS, IRS, I'm telling you. I'm not going for the lower side of jobs. 1000 is not a less in numbers. Okay, so just wait. Okay, time will come, don't worry. I'm giving you people have a proper time. In the last I tell you na, even I discuss your all the questions. I want to see every questions. Okay, this is the reason I said. Because I want that you people should feel I'm taking your live classes. Okay, that literally I'm in front of you. I never want to avoid a single questions. Just that's why when I see somebody are asking me a questions, when I start to give an answer, so I have seen that somebody has given the answer. So it's good that people are helping to the other one. It's very good nature. Okay, but I thought that nobody's are giving the response and all. Then I start to give my answers. So this is the reason if single person will say something wrong, something is here. So I totally distract. Okay, it's my nature because I want to take everyone together. I never want to do that. You people are asking me a questions. I am busy in my studies and teaching. No, because I came here especially for you. So my motive is to connect with you. Okay, I know guys, this time I cannot see you. And that is the reason I told you for the all live classes. I will come to the Mangaluru and I will love to meet you if you are in my nearby areas. Literally I want to see the faces because many people are talking to me. I said, but yeah, I cannot see you. Who are you actually? Okay, so I hope you understood. Sometime I'll get it. Even many places I go. Sometime in the airports or somewhere. Many people said, hello sir, how are you sir? Okay, many of the crew members, they give some plate, sir, please take a coffee. Arey bhai, why I'll take a, sir, I'm your student, you don't know. So I feel good. Recently it happened, I was on the SpiceJet flights and my luggage was I think 5 kg more weight because I was ready to pay. Okay. But the guy was there, he said, sir, don't worry, 2006 batch. You taught me somewhere. So don't worry, sir. I know you very well, sir. If any kind of help from my side. So whatever I have done in my past, today I'm earning. Okay, I did not say I was ready to pay, but the guy is not taking the monies and all. Okay, I'm just telling you, it's a respect. Money, now I can bear the 1000 rupees is what I can give it. But the thing is respect. Those I earn. The belief, those I earn. So trust me. Okay, I still not become as a professional teacher. The problem is it. If I'll be the professional teacher, I will avoid your questions. I never want to do it because I came here to connect with everyone. I did not came here to earn the money because every teachers are doing this earning and all. But I want to do something different. Okay, bacha party. Okay, don't worry. Even you just see. Okay, you take me as your best friend. I always told you. It's my nature always. Even I am the student of Kalam sir. Okay, so I know very well how he taught in the class. He make a very funny environment in the class. Okay, but I'm not in front of you face to face classes. Neither I literally want a copy of that guy because he want to care us. That's why we people start to trust on him. Okay, he just sit with next to me and just say, hey, Ravi, what are you doing? Something like, it was the nature of that man. That's why we people feel very comfortable with that teacher in our class. Okay, we will wait on their own classes always. Hey, sir will come and take the classes. Sodha sir is a IT director, IT Delhi director earlier. Okay, I got a quality teacher in my life and I just like to copy them. Okay, copy them. So don't worry. Okay, I want to do something for you people guys. I want to take your examples in the coming years. Hey, I taught of Mr. XYZ, that many people are there. Okay, hey, it was this in 2021 in the live class. In that time, the people are suffering COVID. But that student listened me and finally he got 10th rank. And now he's posting in nearby my areas in Uttar Pradesh where I stay there. And many times he come and meet me and we people enjoy our pastime. How will he spend and all. Okay, so I just want it that I want to see you. I want to take your example in the coming batches. Okay, so if you want that I take your name in the coming batches, so you should have to do hard work. Okay, and be confident. And as a best friend, I'm always in your life. Don't worry. Either you select or now select does not matter. I'm always with you. Okay, fine. Ab now come here, guys. ठीक है Jay, we will meet it. You are Noida, so we will meet it. Okay, but you come first here. I just narrate two lines of the RNAi. Okay, RNAi is a procedure where we are going to do interferences of RNA process. RNA process means post-transcription process. RNA interference, even within the name already features are given there. We are going to interfere RNA mechanisms. Interference means we are going to interfere RNA mechanisms. And what is the RNA mechanism? Post-transcriptions. Okay, RNA mechanism is what? Post-transcription. So ultimately post-transcription mechanism we are going to stop. It's called RNA interference. Interfere RNA mechanism. So what is the RNA mechanism? RNA to all is what? Post-transcription. The post-transcription mechanism is stopped. Okay, fine. So colors. A biotechnology is defined on the basis of different colors. Okay, just like green biotechnology. Actually, you know, guys, this biotechnology in the symbol, it is B and T, capital B and capital T, capital B and capital T. Green biotechnology, white biotechnology, red biotechnology, blue biotechnology. You know that there is many of purple, you know, maroon, what, magenta, different type of biotechnologies. But these four are the main biotechnology color wise. Color wise means applications. Arya, don't worry, okay. I also don't like to left anything. I'm taking proper classes. And the best part of Vinay sir, director of engineering, he told me, Ravi sir, you take proper classes. Whatever the student want, you fulfill their own demand. Okay, that students will feel literally they learn something in the inside. They have not done the time pass. And it's also not my nature, guys. So the same frequency people met here, and that's why we are trying to do best. And definitely you will get this result also. I think in this 15-16 days session I have taken around 6-7 days more left, because this is the last lengthy topic. Okay, now the rest of the all topic will very fastly it will finish. 2 days I'll take hardly miscellaneous topic. Now the rest of the all topic finished. Okay, so I've just told you guys, next week by Friday or Saturday my syllabus will over. Okay, so and see, you people should remember the same frequency people survive and met. So me and his frequency is 100% same, and that's why I love to do work here. Okay, and the work result is what? You people selections. And I always spoken you that I am a good teacher. It will be not proved from my words, it will be proved through your selections. When you clear UPSC, you will write that teacher even helped you, and because of him you people have able to solve the science and tech in the prelims and mains, your environment in the prelims and mains. So I want it. Okay, fine. I think it was enough for motivation also. Okay. So come. So green biotechnology. So biotechnology, capital B and capital T. In this way we are using green biotechnology, application of biotechnology in agriculture. Okay, uses in agriculture. I'll discuss with you like flaccid, tomato. Many of the example of RNAi. It is also the part of green biotechnology, because everywhere we have done something in the sector of agriculture. BT crop, another example, golden rice, many other example I'll discuss. Wait. Okay, many other example. White biotechnology. So green biotechnology, agriculture. White biotechnology. Importance of biotechnology in industry sector, industrial sector. That is called as a white biotechnology. In the industrial sector, it is called as a white biotechnology. Okay, white biotechnology. Red biotechnology in medical sector, in medicine. So application of biotechnology in the medicine is called as a red biotechnology. And a lot of the oceans, diversities and all, that discussion, that study of biotechnology in this ocean, their own diversities, their own many, you know, organisms, characteristics, that study is called as a what? That study is called as a blue biotechnology. Okay, that study is called as a blue biotechnology. So this is the different color wise the study of biotechnology. You remember, green, white, red, blue. These are the biotechnology. Now come. Now directly I'm coming into the some topics. There are 4 topics here. Those are connecting together. What are the 4 topics? See, what are the 4 topics? The one that is called recombinant DNA technology. Recombinant DNA technology. It is in the short called as a rDNA technology, small r. It is in the short called as a rDNA technology. So this is in the short called as a rDNA technology. So this is in the short called as a rDNA technology. So this is in the short called as a rDNA. So recombinant DNA or rDNA technology. Next, transgenics. Yeah, transgenesis. You can also use the term transgenics. Yeah, transgenesis. Transgenics or transgenesis. Transgenic plants, transgenic animals, transgenic organisms. That discussion I'll do. The next one is a genetically modified. Genetically modified organisms. So next one is a genetically modified organism. Next one is a genetically modified organism. Next one is a genetically modified organism. Okay, next one is genetically modified organism. Genetically modified organism. Yeah, here you will get a GM crop, you know, BT type. That example will come there. And one more example I want to tell you, that is called biopesticide. Biopesticide. That you know very well, that is called BT crop. Because BT crop is a result of some of the technology here. So these 4 topics, those are interconnected. And as in the beginning guys, concept wise, all 4 topics are important and I will discuss here right now. So all these 4 topics are important and here I will discuss right now. So recombinant DNA technology, transgenesis, genetically modified organism, biopesticide, BT crop, right? So biopesticides, a BT crop. So biopesticides are BT crop. Actually, you know, this recombinant DNA technology. Okay, this recombinant DNA technology is overall, it is called as a heart and soul of biotechnology. It's not written anywhere. I use this term. Recombinant DNA technology is literally, it is a heart and soul of biotechnology. It is a heart and soul of biotechnology. So recombinant biotechnology is what? It is a heart and soul of biotechnology. So it is a heart and soul of biotechnology. One point. It is a heart and soul of biotechnology. What will come next? Actually, you know, guys, when the recombinant DNA technology developed, a big revolution came. Okay, and later on the paper started genetic engineering and biotechnology. But one more point I want to tell you. If you go the background of the plants and its cultivations, okay, all the topic I'll come there. But before it, I want to give you a broad perspective. If this type of question suppose come in your essay, so that all entire studies will help you to write a essay. Even those discussion I am doing in the class, even same I'll do in the class about AI. Maybe tomorrow, yeah, day after tomorrow I'll reach there. AI, robotics and nano. Here during the discussion of AI, whatever the discussion I'll do, it was a essay question in 2019. 2 essay in your paper UPSC asked. The one essay, most of the people, those even listen my classes, what about the story and technical part? Story and technical part, I define is AI, artificial intelligence. Many people have written that essay. So science and technology somehow will also help you in your essay writing. But I'm not taking the classes for essay. So, but somehow I want to give you some historical background also. Actually, the biotechnology type of thing started in the respect of the cultivation of the plants from the beginning. Improve varieties. Those hybridization people has done. A good breed and the another good breed, they fuse together. Ultimately, some good genetic character they start to form. If the parents are good, so what about their own breedings will be good. Even Gokul mission, embryo transfer technology, I'll discuss there when I will continue to the some reproductive part. ETT, embryo transfer technology, Gokul Gram and lot of the projects. That discussion I'll do. During the discussion of surrogacy, I will be there. Mahiwal, Seri, many of the Indian breeds that we want to enhance it. Or the genetic breeds and all. So ultimately that hybridization and all the process from the beginning, when the people started to do the cultivations. Actually 3 terms I'll tell you how the plant started to do cultivate. You just listen. It's some background. Technical part, you just wait. I'm coming. Okay. One term is domestication. One term is called as a domestication. Second term is called as a introduction. And third term is called as a acclimatization. So 3 terms here I'll explain. The one is a domestication, second is a introduction, and third one is a acclimatization. So 3 terms here I'll discuss. The one that is called domestication, second is the introduction, and third one is the acclimatization. What are the domestication, introduction and acclimatization? Listen guys, what is domestication? Actually all those plants people can see in your surroundings, all those plants you can see in the surrounding. In the beginning all these plants was in the wild form. In the beginning all these plants was in the wild form. They were growing somewhere, but the people never knew that importance. People never know about its importance, and people never start its cultivations. In the beginning it was in the wild form. So in the beginning every plant was in the wild form. In the beginning all the plants was in the wild form. Okay everyone, it was in the wild form. What happened? Slowly and gradually people understood the importance of these plants. Okay, slowly and gradually people understood the importance of this plant, and they have started to domesticate it. Domesticate means they started to cultivate it. Domesticate. Okay, and see guys, in this cultivation, when the people started to grow of these plants, in these plants, those seeds and varieties are good. Their seeds they save and then further grow. So in the beginning the better quality was improved. It was on the basis of normal procedure. It was called as a selections. So those seeds and all they eat up, they got better, they grow that plant. Other seed was not good, they refuse. And further the seeds they grow, again in that those seeds come, again those seeds have a better quality. Though after few generation, the genetic characters even improved. But here directly they are not going to focus the genetic character. Okay, they have adopted the outside of characteristics. Internally they never know what is the gene and what is the gene. But from the outside of characteristics, finally they were improving the gene indirectly. Not directly, indirectly. And this is what, and later on the plants even domesticated and people start to cultivate it. So what is the domestication? Domestication is the process through a wild characteristics can start to be cultivated as a domestication. So domestication, I hope you people clear. A plant start to domesticate, it's a process of domestications. Okay, and what about the procedure? In the beginning scientists have adopted, the people have adopted selection, normal selections, nothing more. And after certain year to year, generation to generation, because of selection, now finally the variety improved. This is called domestications. So domestication means the plants that was earlier wild, now it start to domesticate for cultivations. And those areas where the plant firstly wild to domesticate, that area become as a what? Center of origin of the plants. That is called center of origin of plants. Okay, you know this plant is this origin. Why so? Because first time this plant was cultivated in these regions. After this domestication, what happened? This plant even moved from one place to another place. That is called as an introduction. So introduction means what? That plant is going to introduce from one region to another region. So what is the introduction? The plant is going to introduce from one region to another region. So domestication, plant is going to introduce from one region to another region. This is domestication. So plant is going to introduce from one region to another region. This is called domestication. This is called as a domestication. Okay, this is called as a domestication. So I hope you people very clear what is the introduction, domestication, what is introductions. I can give you one example. After introduction, plants can start to grow in another country also. Just like I can tell you one example, guys. Tea, you know, tea, chai. I too love tea, guys. Coffee, okay, but tea love it most. Those tea we are taking it, guys. It's a primary center of origin is the what? China. It means first time this tea was started to cultivate in China. Yes. After it, guys, this tea has moved to other countries. Introduction. And India is also the country where the tea came, and tea India become as a secondary center of origin. And how the India becomes a secondary center of origin? After introductions. I hope you know very well what happened here in the case of introductions. Okay, what happened here in the case of introduction? Everybody clear? Introduction. What is the acclimatization? Acclimatization means after introduction, this plant stay there, establish there. That is called acclimatization. Adopt there. So when after introduction a plant is going to adopt, when the plant is going to adopt to the particular area, a region, then that is called as a, now this is the acclimatization happen. But in this beginning, entire worldwide those plants started to cultivate. It was a procedure, and finally here what happened? In this procedure we have also used what? Improvement of the plants. Ultimately, genetic character also improve. And those plant improve, they survive. Rest of the plant remove and die. And not started to cultivate. Still they are in the wild form. And similarly, the same thing we are also doing here, but everything we are doing into the another way. So this is the reason when you people start to the history of the biotech, the story starts from this type of discussions. And few terms was required like domestication, introduction, acclimatization. So I just used here. Okay, I just use here. Come here next, guys. Yeah, I was talking to you about the recombinant DNA technology. It's a heart and soul of biotechnology. Recombinant DNA technology, it is what? It is a heart and soul of biotechnology. How it is the heart and soul of biotechnology? Question is, how it is the heart and soul of biotechnology? Listen, guys. Actually, to come on the recombinant DNA technology, first of all, I'll tell you when restriction enzyme was discovered. When restriction enzyme is also called as a restriction endonuclease. Restriction endonuclease. It's a very specific enzyme or chemicals, I can tell you, which have a catalytic property. It work as a Caesar. Caesar means, you know, catchy. Caesar. It work as a Caesar. This enzyme was discovered by Arber, Arber, and he got this enzyme in E. coli bacteria. E. coli is bacteria where the Arber firstly identified this type of enzymes which work as a Caesar. And this enzyme discovery, restriction enzyme or restriction endonuclease, in the short it is called as a RE. Okay, because of it, guys, recombinant DNA technology become possible. So those recombinant DNA technology, I'm telling you as a heart and soul of biotechnology, how this recombinant DNA technology becomes successful with the help of restriction enzyme discovery. Who discovered the restriction enzyme? Arber. Where this enzyme was discovered? In E. coli bacteria. And after this discovery of restriction enzyme or restriction endonuclease, now recombinant DNA technology become possible, and because of this there is a revolution in biotechnology. And this is the technology those I'm telling you as a heart and soul of biotechnology. Definitely you will ask me, sir, how restriction enzyme is working? In different way, guys. They cut DNA at the desired places. Restriction enzyme, they cut DNA at a specific site. They cut DNA at a specific site. If you can ask me, sir, what is the importance of it? They cut DNA at a specific site. Suppose, guys, this is DNA. Suppose this is DNA. Suppose this is DNA, this is DNA. A T A C G A C C C T, okay, A T A T G C T G G A and T, DNA. It's a DNA. Okay, it's a DNA. Suppose a very specific restriction enzymes, EcoR1, many name, but okay, don't go on the types of restriction enzyme. And this restriction enzyme cut DNA between A and T sequence. So where is the A and T sequence? Here is the A and T sequence. It will cut here at this point. Where's the A and T sequence? It will cut here in this point. And when the DNA cut at this point, so how DNA will cut? DNA will cut like this. DNA will cut like this. DNA will cut like this. And finally, what about the result? This portion of DNA separate and this portion of DNA separate. So it cut DNA at a specific site. Okay, and now what we can do? This is the DNA sequence those are going to cut. We can use it and transfer it to another plants, another organisms. And this type of space. So actually, guys, I want to give you theory, because in this way mechanism will not come. What is the use of restriction enzyme? We can cut a specific DNA sequence. Okay, now this DNA sequence we can remove outside. Yeah, we can take from the cutting of DNA sequences from organisms and transfer to the next organisms. So which enzyme is used to cut the DNA segments in the whole DNA set? Restriction enzyme. Of course, if suppose somewhere have a defected gene, so you will cut that DNA sequences from entire genomic sequences or not? Yes, restriction enzyme you can use. Yes, somewhere you want to take a sequence from one to and transfer to the another one. Again here restriction enzyme used. Again here restriction enzyme will use. Okay, so ultimately what is the use of restriction enzyme? It cut DNA at a specific site. And because of this, somewhere of the gene DNA sequences we will take and somewhere we can transfer. I'll prove it, guys. I will prove it. And what is a recombinant DNA technology? It's a technology through that we can transfer a gene from one to another organism. This is the technology those we are using. So through the help of recombinant DNA technology, we can transfer a certain gene from one organism to the next organism. Okay, through the help of it, we can transfer some gene from one organism to the next organisms. How it is so, entire discussion I'll do in the case of recombinant DNA technology. Okay, recombinant DNA technology. See here, guys, in this case. Recombinant DNA technology, what happened? Suppose one side I am telling you some carrier or vector. Some carrier or vector is used. You just see what I am doing. After some time you will get the answer. Some carrier or vectors people are using. Through the help of carrier or vectors, we are going to carry a very specific gene. Okay, fine. You give me an example, guys. Do you know about the biopesticides? BT crops. BT crops. What is the story of biopesticides? Actually, lot of the plants, they even infect, they were destroyed, damaged because of some specific insects. And that insect was bollworm. That insect was bollworm. You just see, guys. The insect was bollworm. So insect name was bollworm. That you can also call as a white fly. White fly. It damaged a lot of the plant worldwide. So bollworm or white fly, it expelled, lot of the plant worldwide. And guys, what is the problem here? These plants was economic important plants. These insects belong from a very specific group of the insect that is called Lepidoptera. Lepidoptera group of insects. So bollworm and the whitefly belongs from Lepidoptera group of the insects. Somewhere maybe Lepidopterians, in that way example can come. So don't be confused, it's a right. That human is coming what? Okay, that human is coming what? Okay, human is coming in this examples. Just a minute, guys. Okay. So bollworm or the whitefly that belong from the specific group that is called Lepidoptera. Actually, this insects destroy a lot of the plants worldwide. Destroy a lot of the plant worldwide. Up to kill these insects, farmer used pesticides. And the amount of pesticides, okay, increase because insect got resistance. Again, insect got resistance. So ultimately, what happened? The amount of pesticides level increase, and that was very dangerous for everyone, for the environment and human being. Insect got a resistance. Further, the dose of chemical substance increases. Further, insect got resistance. Further, the dose of chemical substance increases, and finally, they are going to hit to the living organisms, environment. Effect of pesticides. Up environment, biomagnification, what is the odd body, cancer, lot of the things. Okay. Up now, this is not safe. What we should do as a next alternative, BT crop form, biopesticide form. What is a biopesticide? Actually, some of the genes, and the interesting, that gene we got from bacteria. Some of the those gene we got from bacteria. Okay, that is called Cry1 AC gene. Tomorrow I'll discuss Cry1 AC gene. This was the gene. This gene was identified in a bacteria. That bacteria, I hope you know very well, it's a soil bacteria, gram-positive bacteria, Bacillus thuringiensis. From this bacteria, scientists has observed. And this gene from the Bacillus thuringiensis, we have taken it and transferred it to the desired plants. Desired plant means like tobacco, like brinjal, like cotton, and so many of the potatoes and a lot of the plants. Okay, and now what happened after the transferring of the gene? Now some protein formation start into the plants. Those plants got infect Yeah, destroy because of what? This bollworms or white fly. Now when the insect will eat bollworms and all, what will come there? It will affect their own gut parts. Gut means alimentary canal wala part. Blockages happen, and now the insect die. So those insects earlier die because of chemical insecticides, pesticides. Now because of plants, because of some genetic character when we have changed, plant already synthesize some toxic substances. And that toxic substances, when the insect will eat up like bollworm, they die. And now here plant already got resistance against the insects. Insect means what? Bollworm. And this plant itself called as a biopesticide. Plant itself become as a pesticides. Resistance against the insects are pest. Again the insect are pest. This is what? This is biopesticides. So what actually biopesticide, this mechanism I will discuss tomorrow. But in the case of formation of biopesticides, ultimately we have taken this procedure. And behind this procedure was a need of restriction enzyme. And somehow recombinant DNA technology connecting with the transgenic and genetically modified. And this is one of the example. Because of shortage of time. It's 9:59. I cannot discuss, but it will take around 30 minutes topic. At least 30 minutes. Just like today I discussed with you about the division, cell divisions, some discussion I have done in the previous lecture. Okay, fine. So, do you have any doubts? It's your time. Do you have any doubts? You can take right now. Rest timing not allowed. It's an entire connecting topic because recombinant DNA technology connecting with the transgenic. Transgenic connecting genetically modified. Genetically modified example is a biopesticides. And importance of genetically modified plants tomorrow I'll discuss. So it's entirely 1 hour, at least 1 hour discussion at this stage is required. So no questions. Well, people were waiting to, sir, you say I'll go. So I think the basic parts that was literally very, very important. Everybody clear? Now if UPSC will turn any questions, so now you can guess very easily. Bacha, those form I told you. Agar blue rose not come, purple color, the purple I'll tell you. Ab it's a part of research work. How to read the environment? Environment what? TB. What is environment TB? How, sir, how to read the environment TB what? As a textbook. What TB? Textbook. You can ask any doubts, don't worry. But my 2 subject based doubt only you can ask me, not other. Okay, when I'll come to the Bangalore, okay, you can ask me, don't worry. These 2 are my subject, my favorite subject. Even first of all, I'm the environmentalist. Reason the environment subjects, everybody's are taking classes, okay. So science and tech, I got more popularity, name, fame and all. Okay. Acha, textbook. So nothing, you just follow the, actually in the case of textbook, I'll tell you one thing. After every units, one day I'll take one session, especially for one session means hardly 15-20 minute discussion to how to go with the books. Don't worry, if you have any need of it, so I'll go it. Tomorrow if you say, so tomorrow I'll tell you, but better if you'll have a book, so in that case I will help you how to go it. Okay, so tomorrow I'll tell you. Okay, I'll take a last 50 minutes especially for it. Okay, but first of all, yeah, better last day you ask me. Okay, it's my humble request. I'm here for the science and tech, so you guys ask me science related questions. Okay, if you people met me anywhere, so in that case it's fine, but here you ask me science and technology. Okay, you have a book, so don't worry, I'll tell you how to do these studies and all. So thank you everybody, and have a good day. Bye. Why you are going for vaccine manufacturing process and all? यार क्या कर रहे हो तुम लोग? Okay. आप मतलब scientists could not develop the COVID vaccines, but you will develop first vaccine. Okay, COVID vaccine. अच्छा. And definitely I will make the COVID vaccine with you people here today. And next day we will get the Nobel Prize all. Okay. Now what you people are doing? Okay, fine, whatever. Thank you everybody. Have a good day. Bye.